everyone, welcome back to channel GDC English. So here we are back again for you guys with UPSC Drug Inspector 2023 PYQ's explanation. Okay, so I hope your preparation is going good and you are also going good with your preparation, right? So momentum has started and here we are with boost up for your momentum so that you can carry forward ahead. Okay, so here in this one, this is your UPSC Drug Inspector 2023 question paper with explanation, guys. Hana? So let's go ahead. And if I talk about pharmacology and allied subjects, okay, total how many questions they have asked over here? That is around 30 questions they have asked in 2023. Okay, how many from that? General pharmacology, 3 questions. Autonomic nervous system, only 1. Autocoids, 2. Respiratory, nil. Hormones and related drugs, 7. See, maximum they have asked 7 from hormones and related drugs. Drugs acting on peripheral nervous system, that is 1. Central nervous system, 4. Good one, right? Then cardiovascular drugs, 2. Kidney, nil. Drug affecting blood and blood formation, 4 questions they have asked, right? After that, gastrointestinal drugs, that is your 1. After that, antimicrobial, 4. Neoplastic disease chemotherapy, say they didn't have asked any question. That's a strange one. Miscellaneous, they have asked 1. Total, they have asked 30 questions over here in pharmacology allied subjects. So through this, you can get an idea that how many questions they have asked from each of the chapters. So in that perspective, you should be focusing more onto those chapter, right? Okay, so this is your pharmaceutics, guys. Hana? Pharmaceutical technology, 19 question they have asked. Physical pharmacy, 0. Biopharmaceuticals, 9 question. Hospital dispensing, 0. Cosmetic, 2. Jurisprudence, 36 question they have asked. Engineering, 0. Total 66 questions. 66 questions they have asked from pharmaceutics and allied subjects. Okay, maximum weightage here you can see over here. All right, in pharmaceutical chemistry, here you can see organic chemistry, only 4 questions. In organic chemistry, 1 question. Analysis, very important, 13 questions. Medicinal chemistry, 5 questions they have asked. Total 23 questions they have asked in pharmaceutical chemistry and allied subjects. All right. Okay. Pharmacognosy, if I talk about the introduction of pharmacognosy, 0. Alkaloids, 2. Lipids, 1. And rest of the chapter also 0. Total question they have asked, that is your 3. Okay. Next. Okay. Biochemistry, carbohydrate, 2. Vitamins, say they have asked 1. Total 3 questions they have asked. In microbiology, 9 questions they have asked. 1 from sterilization, 5 from immunology, 3 from vaccines and sera, and total is for 9. All right, this is important. Next, non-pharma 13 questions they have. 6 from the computer, 5 from English, 2 from current affair, and 0 for reasoning. That's a strange. Okay, fine, guys. Now let's move on to those questions that we have for your pharmacology and allied subjects. Okay, so let's start this one. Okay, so the first question says that which of the following protein is mainly responsible for binding with drug in plasma? Which of the following proteins is mainly responsible for the binding with drugs in plasma? Apolipoproteins, albumin, transferrin, or immunoglobulin G? What's that? Yes. See, many of the drugs do bind with plasma protein. Plasma protein, we do have albumin, we do have alpha-1 acidic glycoprotein, we do have that globulin, we do have hemoglobin, we do have lipoproteins, right? But maximum of the drugs are going to bind with those albumin only, right? So albumin is going to be your correct answer. Okay. So acidic drugs are particularly bind with your plasma albumin, and basic drugs particularly binding with alpha-1 acidic glycoprotein. Here is a one trick I would say through which you can determine these are the basic drugs. What are those? What are those? Here you can see bupivacaine, E at the end. Disopyramide, E at the end. Imipramine, E at the end. Lidocaine, E at the end. Methadone, E at the end. Quinidine, E at the end. Maximum of the drugs, those who are binding with alpha-1 acidic glycoprotein, only basic drugs are there which are going to bind with alpha-1 acidic glycoprotein, right? So the drugs which is having at the end E except 𝘿𝙄𝙎𝘾𝙄𝑖𝑑E are considered to be as your basic drugs. Other than these drugs, we do have plenty other basic drugs as well, acidic drug as well. For the time being, or you can say for this table, the drugs at the suffix at the last alphabetical order, E must be present over there, then it will be binding to alpha-1 acidic glycoprotein. That is your, because that's become your basic drugs. All right. Okay, fine. Let's go ahead now. Pre-systemic metabolism. Pre-systemic metabolism is also known to be as what? First pass metabolism, right? It is also known to be as your first pass metabolism. What is first pass metabolism? First pass metabolism is nothing but when the drug is going under the metabolism before even going for absorption. Even before they are going for absorption, that is what? Pre-systemic, or you can say first pass metabolism. And that is usually happens to those drugs which are being given through oral route. Maximum first pass metabolism leads from your oral route and leads towards decrease in efficacy, decrease in bioavailability. All right. Now, these options. Pre-systemic metabolism occurs in liver as first pass metabolism. Yes, right. Occurs in intestine and swallows. Yes, that is also right. Occurs when drug is administered transdermally or inhalation. No, I told you first pass metabolism maximum occurs with what? With your oral route. Okay, now result in decrease in bioavailability. Yes, so 1, 2 and 4 is going to be your correct answer. Yes, 1, 2 and 4 is going to be your correct answer. This is your correct one, correct option. All right. Chala, fine. Let's go ahead now. Now, which one of the following types of antagonism is observed between the actions of nitrites and cyanides? Okay, antagonism is between observed between the nitrites and cyanides. So you know that cyanides may cause what? Cyanides may cause cyanide poisoning. है cyanides may cause the cyanide poisoning, but the antidote of that cyanide poisoning is your sodium thiosulfate, nitrites, right? These are your, and you can say antagonist, है antidote as well. So what kind of antagonism does it actually takes place? Physical antagonism? No. Physiological antagonism? No. Chemical antagonism? Yes. Chemical antagonism generally takes place with nitrites as well as cyanides. How? How? Here you can see that cyanides are being going to interact with cytochrome C oxidase. Cytochrome C oxidase. Okay, cytochrome C oxidase, and which is in oxidative high phosphorylation, which happens in oxidative phosphorylation, right? That is your ETC chain, electron transport chain, electron transport chain, right? Electron transport chain, correct? Now, once this cyanide is going to bind through with oxidative phosphorylation, that is electron transport chains, what's going to be happen? Cytochrome oxidase C, that is going to metabolize. After that, what do we have? This leads to what? This leads to your cellular hypoxia. Cellular hypoxia and respiration may slow down. Respiration may slow down, right? But when nitrites come, these nitrites convert hemoglobin converts hemoglobin into methemoglobin. Okay, converts into methemoglobin. What's going to be happen? Hemoglobin converts into methemoglobin. Now this formed methemoglobin is having higher affinity towards cyanides, higher affinity towards cyanide. So what's going to be happen? Cyanide going to left the binding of cytochrome C oxidase and start binding towards this methemoglobin. So hence what? Cytochrome oxidase starts recovering, and this hypoxia will be also recovered. So this is how the nitrites used in your cyanide poisoning. I hope it is very much clear to you all. No, no, no. This is not competitive antagonism. This is your chemical antagonism, guys. Chemical antagonism. Nitrites binding to and neutralizing cyanides. Here you can see chemical antagonism. That was wrong. Now, which one of the following is an example of angiotensin 2 blocker? Angiotensin 2 blocker? Angiotensin 2 blocker. What's that? Angiotensin 2 blocker. Angiotensin receptor blocker, I know. Angiotensin receptor blocker, I know. But what is this angiotensin 2? Fosinopril, clonidine, losartan, or propranolol? What do you want to answer? ARB they are. Okay, angiotensin receptor blocker, right? So this is your losartans. All the sartans category comes under the angiotensin receptor blocker. Fosinopril, that's your ACE inhibitor. Clonidine useful in cheese reaction, right? Many of the reactions, right? Also, clonidine is your alpha 2 agonist. Propranolol, that's your non-selective beta blocker. Okay, low sartan. Angiotensin 2 receptor antagonist. Okay, angiotensin 2 receptor hai wo. Now, which one of the following antiarrhythmic drugs produces brief therapeutic action and transient adverse effect? Brief therapeutic effect is being seen only by the drug which is known to be as diltiazem, propafenone, esmolol, or adenosine. That is your adenosine over here. D is going to be the correct answer. Okay. Produces transient dyspnea as well as chest pain, fall in BP, flushing. Okay. Now, which one of the following statement regarding the phase 2 clinical trial is correct? Phase 2 clinical trial. So what's that? Phase 2 studies are also called to be as your pivotal trials. Phase 2 study in primarily done to determine the toxicology and pharmacokinetics of the test drug. Okay. Phase 2 study is the first controlled clinical study done to determine the efficacy and safety in patients. Phase 2 study begins with the submission of a new drug application. A new drug application that has been done after phase 3. So D though automatically not. So which is going to be your correct answer? That is your C. Phase 2 study is the first control clinical study done to determine the efficacy and safety in patient. Okay. Fine. Chalo. Next. Okay. So this is your batch, guys. Drug inspector free batch test series. Daily practice tests are being given over there, guys. So you can go ahead with that, right? And you just type GDCDI into the batch as a batch code and you will be enrolled into the test series. Okay, batch. Fine. Here you can see install the GDC classes app, batch section, give it a plus icon and then enter batch code. That's your GDC DI and then you will be good to go for that. Okay. Now, thalidomide previously banned for its teratogenic effect. What's that effect? That's your phocomelia. And is again introduced in the market for which of the following? For which of the following? It is now banned. It was previously banned for something, but it is now came into your market. For what purpose? Sedative, hypnotic, anticonvulsant, lepra reactions, or antibiotic. What's that? That is your lepra reactions, guys. For lepra reaction, for leprosy, they have been introduced again into the market. Okay. So, all of the following drugs can be useful by diagnosis of myasthenia gravis except neostigmine, D-tuberculin, edrophonium, or physostigmine. What's that? Yeah, neostigmine, D-tuberculin, edrophonium, or physostigmine. See, for the diagnosis, edrophonium and D-tuberculin are being used for the diagnosis. Neostigmine is being used for the treatment, right? If treatment, so that can also be used as a diagnosis. But physostigmine is not being used as your treatment. Okay, not even a treatment, not as a diagnosis. Okay, myasthenia gravis is, you know that's your autoimmune disease. है ना, caused by a auto antibody that is going to act onto the nicotinic receptor at muscle end plate. Okay, this is the thing that you have to remember. Now, burimamide is a full antagonist of which of the following receptor? UPSC DI 2023. H1 receptor, D2 receptor, H2 receptor, or D1 receptor? Burimamide. Burimamide. Burima. Okay. H1 receptor, D2 receptor, H2 receptor, or D1 receptor? That's your H2 receptor. Okay. So fine. Burima receptor. Hmm. Okay. H2 antagonist, burimamide. First H2 antagonist. H3 inverse agonist, that's your triptyline, which has been used in narcolepsy. Narcolepsy is what, guys? Narcolepsy is that condition where you sleeps a lot. That's your narcolepsy. Okay. Chalo. Now, paracetamol was detected as a metabolite of diclofenac, ibuprofen, ketoprofen, or phenacetin. Yeah, paracetamol was detected as a metabolite of what? Diclofenac, ibuprofen, ketoprofen, or phenacetin. Very simple question. Paracetamol is your what? Paracetamol is the active metabolite of your phenacetin, right? Phenacetin is a prodrug for the paracetamol. Once it gets converted into its active metabolite is nothing but this paracetamol only, right? And if I talk about the paracetamol goes towards the glutathione conjugation reaction. Okay, remember that. Paracetamol. Okay. Now, which of the following drug is not commonly combined with methotrexate for the treatment of rheumatoid arthritis? Okay, not generally being combined with methotrexate. Sulfasalazine, leflunomide, azathioprine, or chloroquine? That's a good question, I would say. And because all these drugs are being used in your rheumatoid arthritis, but which should not be used as a combination with methotrexate. Sulfasalazine, leflunomide, azathioprine, or chloroquine? That is your chloroquine, guys, that you should not be given over here. Okay, sure, fine. Pegvisomant is used for the treatment of which of the following clinical conditions? Pegvisomant is what? Pegvisomant is your growth hormone receptor antagonist. Growth hormone receptor antagonist. Okay, that's your pegvisomant. So it has been used in your infertility, pituitary dwarfism, acromegaly, or Turner syndrome. What's that? See, growth hormone is responsible for what? It's responsible for increasing the growth of the cells, of the tissues, right? Hana? So, what's going to be happen? If in that case growth hormone exceeds, in adults acromegaly takes place. In child, gigantism takes place. And growth hormone, if remains down, at least the dwarfism, which is also known to be as Lorraine-Levy syndrome, right? But this pegvisomant is being useful for the treatment of acromegaly. Acromegaly is what? Increase growth hormone levels in adults. That's your acromegaly. So to decrease down the effects of growth hormone, pegvisomant has been used for your acromegaly. Okay, fine. All right. Now, which one of the following anti-diabetic drug is mainly given to control postprandial hyperglycemia in type 2 diabetic mellitus? That means what? Postprandial means after meal glucose shoot, right? So after meal glucose shoot may which drug is being used over here? Metformin, pioglitazone, neteglenide, or dapagliflozin? Metformin, AMPK activators, biguanides, pioglitazone, thiazolidinediones. These are the two drugs which are being used to increase the insulin sensitivity and decrease the insulin resistance, right? Neteglenide acts same like sulfonylureas and a potassium channel blocker, also going to enhance the release of insulin. Dapagliflozin, that's a SGLT2 inhibitor, newer drug. SGLT2 inhibitor, sodium glucose linked transporter, right? So over here, if I talk about, so neteglenide becomes the correct answer, which has been mainly used for your hyperglycemia postprandial. Okay, all right. Now, Which of the following antidiabetic medicine undergoes metabolism by cytochrome P450 enzyme and has drug interaction with rifampicin? Rifampicin, you know? Rifampicin? Rifampicin, that's your anti-tubercular drug. Acarbose, metformin, pioglitazone, or sitagliptin. What's that? Hmm? That's your pioglitazone. Pioglitazone, that's your thiazolidine which are going to increase the insulin sensitivity, right? So this has a drug interaction with rifampicin. Okay, this is the thing. Rifampicin is a CYP2C8 inducer. That means enzyme inducer. That means enzyme inducer, right? And so the increased metabolism of pioglitazone takes place. Enzyme inducer, you know, there is one drug, there is another drug which are being given in combination. Hana? So there is a one enzyme inducer. Inducer means stimulates the metabolism of other drug. So if the metabolism of other drug is going to be increased, what's going to be happen? Efficacy and bioavailability is going to be decreased down of that drug, Hana? Leading to reduced plasma concentration and decrease the hypoglycemic effect. That's your the thing with pioglitazone. Okay. Now, all of the following drugs acts by stimulating glucagon-like peptide GLP-1 receptor and are useful treatment of type 2 diabetes mellitus except exenatide, repaglinide, liraglutide, or dulaglutide. What's that? Simple. That is coming from the classification only. Exenatide, repaglinide, liraglutide, dulaglutide. That's your repaglinide, guys, which comes under the meglinides category. Okay. So that's your repaglinide is going to be the correct answer. Okay. Chalo, fine. Now, which one of the following monoclonal antibody is specific for rank L and it indicated for the treatment of postmenopausal women with osteoporosis? What's that? It's your eculizumab, palivizumab, ranibizumab, or denosumab. This is your denosumab, guys, which is specific for your rank L and indicated for treatment postmenopausal women with osteoporosis. Okay, chalo, fine. Now, which one of the following is the mechanism of action of bisphosphonates in the treatment of osteoporosis? Okay, so what happens in osteoporosis? There are two cells, you know, osteoclast and osteoblast. Osteoclast are your what? Bone, bone, bone degrading cells. Okay, and osteoblast are your bone forming cells. Hana? So what's that? Osteoblast and osteoclast. So increasing the osteoid formation, decreasing the osteoclast mediated bone resorption, increasing the mineralization of osteoid, or decrease the parathyroid hormone secretion. So what's going to be takes place? This is obviously, as I told you, osteoclast is for responsible for your what? Responsible for your what? Responsible for your bone resorption. That is remove of calcium from the bone, right? This is the thing. So B is going to be the correct answer. Exactly. Okay. Which of the following anti-arrhythmic drugs produces brief therapeutic action and transient adverse effect? Already we have done. Okay, so this question has come twice. This is your adenosine, guys. Adenosine that we have already discussed this one. Okay, now which one of the following statements regarding group O is correct? Okay, so you know that blood group is defined by Karl Landsteiner. Karl Landsteiner was the scientist who discovered this blood group classification of blood group A, B, AB, and O. Right? Positive is defined with the antigen that is present onto it. Okay, antigen. Right? So for A blood group, A antigen is there. For B blood group, B antigen is there. AB, both the antigens is there. For O, there is no antigen is there. There is no antigen, right? So, if I talk about antibodies is just reverse in that condition. For A, B antibody is there. For B, A antibody is there. For AB, there is no antibody over there. Okay. And for O, both the antibodies are there, A and B. Okay. So if I talk about It has agglutinogen A only on the RBC. For O, no. That we are talking about B. It has agglutinogen B only onto the RBC. That is talking about A. Okay. It has agglutinogen A and B both onto the RBC. That is your AB. It has agglutinin anti-A and anti-B into the plasma. That's your antibody, right? And I already told you that's your D. Yes, the D is going to be your correct answer. Okay. Now, if I talk about universal donor or universal recipient, that is also a question they used to ask. Another universal recipient is your AB, but which? AB positive. AB positive is considered to be as your universal recipient. Okay. AB negative, sorry, not AB negative. O negative is considered to be as your universal donor. Universal donor. The O negative donor. है ना, O negative donor. Donor में ही you can see the O negative. Okay. This is the way that you can learn all this thing. Fine. All right. Ceruloplasmin is responsible for binding and transportation of which one of the following elements in the blood? Is it your copper, iron, magnesium, or zinc? What's that? That is obviously your copper only. Ceruloplasmin, copper. Okay. Also, D-penicillamine is being used as an antidote for copper poisoning or Wilson disease. Copper poisoning or Wilson disease. If copper poisoning takes place, then D-penicillamine is your antidote for that. If I talk about copper-induced Wilson disease takes place. Wilson disease is the accumulation of copper in your brain. So, in that condition also, D-penicillamine is being used. Okay. So, now, erythropoiesis is the process of erythropoiesis. Erythropoiesis, erythro means red blood cells, and poiesis means formation. Red blood cells ka formation, that is erythropoiesis, right? Development of red blood cells, development of thrombocytes, development of white blood cells, development of monocytes. Obviously, that is your development of red blood cells. Erythropoiesis takes place. That takes place in your bone marrow, right? Bone marrow. And erythropoietin, which has been hormone, is primarily produced by the kidney only, but the formation of erythropoiesis takes place in your bone marrow only. Okay. So, now which one of the following clotting factor does not require vitamin K during blood coagulation process? So you know that vitamin K is your clotting vitamin, है ना? Clotting vitamin. So it produces clotting factors, right? Is it your factor 9, factor 5, factor 7, or factor 10? Right. The factors are very much important in order to understand regarding those vitamin K. The vitamin K is responsible for the production of factor 2, 7, 9, and 10. Okay. This is you have to remember. 2, 7. 2 plus 7 is equal to 9 and add 1, that is your 10. So 2, 7, 9. Yes. 5, no. 7, yes. Yes, and 10 also yes. So factor of 5 is going to be your correct answer over here. Does not require vitamin K. Okay, so this is going to be your correct answer. Fine. Sure. Factor 5. Now, metoclopramide has the following actions of the gastrointestinal tract. You know that metoclopramide is your prokinetic drug which is act onto the 5-HT4 receptor as a agonist. Also, it has domperidone like action, means dopamine D2 receptor blocker. Except they are asking, it increases the tone of pyloritic sphincter, increases the tone of lower esophageal sphincter, increase gastric peristalsis, increase intestinal peristalsis. Yes, so as I told you that metoclopramide are your prokinetic drugs. These are your prokinetic drugs. So what's going to do? Prokinetic means increase the kinetic, means increase the movement. Which movement? Peristaltic movement, right? Peristaltic movement. So the content that we have in your stomach, that we have in intestine, it moves forward for the defecation process, right? Increasing the tone of pyloric sphincter? No. if you increase the tone of pyloric sphincter, will the food which is present in your stomach, it can be go ahead? No, it can't. If it increase the tone of lower esophageal sphincter, yes, that is one correct. Increases gastric peristalsis, obviously yes. Increase intestinal peristalsis, yes. But A is not going to be your correct answer. Okay, chalo, fine. The following antibiotics are commonly employed for the treatment of Helicobacter pylori associated with peptic ulcer, except, okay, peptic ulcer except if I talk about, so COMT is a trick to remember to for the quadruple therapy, for your quadruple therapy. Okay, quadruple therapy. C for clarithromycin, or you can say ciprofloxacin. Ciprofloxacin. O for omeprazole. M for metronidazole, and T for tetracycline. T for tetracycline. So clarithromycin, ciprofloxacin, yes. And if I talk about CAP, so CAP is for your triple therapy of H. pylori infection. Triple therapy. Okay, triple therapy. So C for clarithromycin. No, not for clarithromycin. C for your CB. Okay, fine. Wait. This is my bad. Clarithromycin or ciprofloxacin are being used over here, over here, and C is over here is your colloidal bismuth substrate. Colloidal bismuth substrate. Okay, substrate. This is your C. A for amoxicillin, and P for PPIs. PPIs, that is your omeprazole, rabeprazole, all those kind of a thing. So clarithromycin, yes. Ciprofloxacin, yes. Amoxicillin, yes. Tinidazole, no. T for tetracycline, not tinidazole. Okay, chalo, fine. Now, primaquine induces hemolysis in some African patients deficient in which of the following enzymes. Okay, primaquine. Hemolysis it can do, right? Hemolysis. Which kind of hemolysis? G6PD deficient hemolysis. What's that? Glucose 6-phosphate dehydrogenase? Yes. Carbohydrates? No. Acetyl cholesterol? No. Chymotrypsin? No. Yes. G6PD deficiency can take place you hemolysis. Okay. This is the thing that you have to remember, right? Now, which one of the following is an example of aminoglycoside antibiotic? Okay, aminoglycoside antibiotic, streptomycin, clavulanic acid, cefaclor, or azithromycin. Okay, yes, streptomycin is considered to be as a aminoglycoside which acts only onto the 30S ribosomal subunit. Okay, it is the only, I can say it is only aminoglycoside. Only aminoglycoside which acts onto the 30S. The rest of the aminoglycoside act onto the 50S ribosomal subunit. Okay. Clavulanic acid, that is your what? That is your beta lactamase inhibitor. Beta lactamase inhibitor. Right? Cefaclor, that's your obviously beta lactam, that comes under your cephalosporins. And azithromycin, these are your macrolides. Antibiotic. Okay, fine. Let's go ahead now. Yes. Okay. Systemic aminoglycoside, topical aminoglycoside, neomycin, and trimacetine. Now, which one of the following is an example of reverse transcriptase inhibitor? Acyclovir, cytarabine, idoxuridine, or zidovudine. Reverse transcriptase enzyme inhibitor. These are those drugs which are being known to be as antiretroviral drugs, means anti-HIV drugs, right? What are those? These obviously are 𝘎𝑉𝑑ine only. Okay, 𝘎𝑉𝑑ine. Acyclovir, that's your anti-herpes, right? Herpes simplex virus, anti-herpes. Cytarabine, idoxuridine, these are your anti-cancer drugs to be precise. Okay, chalo, fine. Which one of the following human leukocyte antigen HLA encodes for major histocompatibility complex 2? HLA-A, HLA-B, HLA-C, or HLA-D? What's that? That's your HLA-D. Okay, HLA-D, major histocompatibility complex class 2. Okay, HLA-D. Now, which one of the following immunosuppressant agent does not cause bone marrow suppression as one of its adverse effect? Okay, bone marrow suppression. Okay, bone marrow suppression can lead towards anemia. Hai. So, which of the following does not cause? Azathioprine, cyclosporin, sirolimus, or methotrexate? What's that? Immunosuppressant they have asked. Azathioprine is your immunosuppressant. Cyclosporin, yes. Sirolimus, yes. Methotrexate. Cyclosporin still have immunosuppressant but does not cause bone marrow suppression. Okay, cyclosporin. Now, which one of the following statement regarding succimer is most appropriate? Succimer is your chelating agent. Hana? Chelating agent also been used to chelate the lead. Lead. Okay. So what's that? It produces less toxicity than dimer-caprol. It is ineffective orally. It is effective for the treatment of iron poisoning. It is contraindicated for use in children. What's that? Most appropriate, I'm asking. Most appropriate. I mean, which is correct? Which is correct? Obviously, this is your A, I would say. It produces less toxicity than dimer-caprol. Okay. Now, okay. Again, last question, I would say. Because one question became twice true, so that's why there is another question. Anti-epileptic drug implicated in causing toxic epidermal necrolysis. Epidermal necrolysis, skin thing, dead of skin cells. Felbamate, gabapentin, lamotrigine, or vigabatrin. Yeah, that is your lamotrigine, which is your sodium channel blocker, over here, and that is one of a drug which has been caused epidermal necrolysis. Okay.
According to GCP guidelines, minimum number of members required in quorum of ethics committee to review the protocol is. So how many members are there in the quorum committee which review the protocol? So basically 5 members are there, which includes your legal expert, clinicians, etc. So options are given 5, 6, 7 and 9. So minimum 5 members are required in the quorum committee, that is the ethics committee. Okay. So 5 is the right answer, option A. Remember, these 5 members are your basic medical scientist will be there, then clinician, legal expert, social scientist and lay person. So these are the 5 members which are there in the quorum committee. Okay. Now you can download the free PDF which is given in the description box, where you will get the UPSC Drug Inspector 2026 complete syllabus PDF is given in the description box. Along with that, you will have previous year's question paper, that is 2015. Direct download link is available, and obviously save this PDF and start your preparation today. Boost your preparation for your UPSC examination. So next question is, according to GCP guidelines, which one of the following statement does not hold true for ethics committee? Basically, which of them is the incorrect statement? Okay. Option A is all members serving in ethics committee that oversees human research at an institution must be employed by that institution. Option B is ethics committee should include at least one member whose primary area of interest is non-scientific. Option C is ethics committee should have appropriate gender representation. Or option D is for review of each protocol, the quorum of ethics committee shall be at least 5 members. So this is the same question from your whatever explanation we have seen for the previous question. So we know that 5 members are there. So option D is the right answer. not answer, the statement is right. Okay, we have to identify which is not correct. Okay. Option C is ethics committee should have appropriate gender representation. That is a correct statement. Option B is ethics committee should include at least one member whose primary area of interest is non-scientific. That means external member. That is true. But first one, what they are telling is it compulsory that all the members should be of that institution? No. One member is of the external committee or externally one member is present. So this statement which tells that it should be of that institute, this is not true. So option A is the right answer. Not required. Rather, he should be from the external background. Okay. Option A is the right answer. The next question is, which one of the following is not considered as the essential element in the study subject's informed consent documents according to GCP guideline? Not considered as essential element. Okay, not considered as essential element. Option A is expected duration of the subject participation. Yes, the duration is important. So option A is not your answer. Option B is an explanation about whom to contact for trial related queries, rights of subject and in the event of injury. Obviously, all the details explanation should be given so that they can contact to the person. B is not your answer. Option C is a statement that there is a possibility of failure of investigation product to provide intended therapeutic effect. Now, this statement does not hold true that this is not a compulsory mandatory thing. It is not there for any kind of documents according to the GCP guidelines. For example, they are doing trials for any kind of investigational new drug application, right? Any kind of new drug is there. So this is not a statement that what if that particular drug is not working and all. So this is not something given. Option C is the right answer. Option D is a statement that a particular treatment or procedure may involve risk to the subject which are currently unforeseeable. Obviously, certain side effects might be there, so you have to make them aware that so and so things can happen. So option A, B and D are the statements which are there, but option C is not considered as the essential element. Okay. So option C is the right answer. The next question is, as per 𝘂𝙎𝙎 6 GCP guidelines, all of the following should be complied except, again, you have to identify which of them is the incorrect statement. That except is there, right? Option is information recorded in the investigator report should be complete, accurate and legible. Obviously, any missed study visit may not be noted in the reports. Subjects who were dropped from or failed to complete the study are noted in the report with the reason adequately explained. Or option D is informed consent should be adequately documented. What should be the answer? Any missed study visits may not be noted in the report. This is not true. Whatever study has been missed, that has to be reported. Okay, it is very mandatory that even the missed study, whichever is there, that has to be reported. So all of them comes under your ICH E6 GCP guidelines except your option B, because they are telling that it should not be noted, which is absolutely incorrect. It has to be reported in the, it has to be given in the reports, or it has to be noted down in the reports. Option B is the right answer. The next question is, as per ICH GCP guidelines, whose responsibility is to ensure that investigators are qualified by education and experience and are trained on the conduct of the protocol? So whenever a study protocol is being conducted, there are various study sponsors are there. Okay. So their responsibility is to ensure that the investigators are qualified, they have their proper education, they have experience in that particular field. So option is given FDA. FDA does not play a role in this one, right? FDA comes afterwards when your study is completed. So FDA is not the right answer. Study sponsor is the right answer. Investigator, how himself he will investigate whether he is qualified or not. So for that, study sponsors are required and not the ethics committee, right? So study sponsor is someone who will make sure that investigators are well educated and well experienced in that particular field, and he or she is responsible for carrying out the study. So option B is the right answer. Very good. Next question is the document retention period as required by FDA. The document retention period as required by FDA following the date of marketing application approval or after the investigation is discontinued and the FDA notified is. So basically they are asking what is the document retention period. Okay, what is the document retention period as required by FDA? Option A is 1 year, option B is 2 years, option C is 3 years, or option D is 5 years. What is your answer? So for the maximum period of 2 years, the retention period is 2 years. Okay, maximum 2 years. So option B is the right answer. So investigators shall retain records which are required to maintain under this part for a maximum, that is a maximum period of 2 years. The next question is WHO guidelines on stability studies recommend storage of pharmaceutical products at different temperature and relative humidity in different zones. The temperature and humidity recommended for zone 4B is okay. So there are various zones depending upon the temperature and humidity. So they are asking what is the temperature for zone 4B, temperature as well as humidity. Okay, 30 degrees Celsius, 60% relative humidity. Your 30 is the right, but 60% relative humidity is incorrect. 30 is right, not 65, not 70. So option D is the right answer. 30 plus minus 2 degrees Celsius and 75 plus or minus 5% relative humidity. If these are the conditions for zone 4B, okay? 30 degrees Celsius plus minus 2 degrees Celsius and 75 plus minus 5%. And minimum duration is 12 months. The next question is when there is a change in the equipment process, there will be need for retrospective validation, concurrent validation, prospective validation, or revalidation. So when there is a change in the equipment of the process and all, you have to do revalidation. Revalidation as in you have to validate that particular process again and ensure that even after a significant change, it is able to produce the desired product or the product with the desired quality. So you have to ensure that the quality of the product is retaining even after the significant change. So to prove that, we will be doing revalidation. We will be doing revalidation when there is a significant change, right? So option D is the right answer. Which one of the following is not correctly matched as per the classification of clean rooms ISO 14644? You have to identify which of them is not correctly matched. Part 1 is given as classification of air cleanliness is the right answer. I mean the statement is right. Part 3 test method, yes, part 3 is for test method. Part 4 is given as design, construction and startup. So that is also right. But part 5 here it is given as terms and definition. Part 5 is given as terms and definition, whereas part 5 is your operations. And part 6 is your terms and definition. Okay. So they are asking which of them is not correctly matched. Part 1 classification is correct. Part 3 test method is correct. Part 4 design, construction and startup is correct. Part 5 terms and definition is not there. Part 5 is operation. And part 6 is terms and definition. So option D is the right answer. And part 2 is for the specifications. Okay. Now next question is which of the following are correctly matched regarding registration of pharmaceuticals for human use? So ICH guidelines basically they have given Q8, Q9 and Q10. What they have given is Q8 is pharmaceutical development, Q9 is quality risk management, Q10 they have given radio pharmaceutical guidelines. You have to identify out of these which statements are correct. I'll give you few seconds and you let me know your answers. So Q8 pharmaceutical development, that is the correct, we know Q8 guidelines is for pharmaceutical development. Q9 is quality risk management. But Q10 is not radio pharmaceutical guideline, it is about the pharmaceutical quality system, right? PQS, it is PQS. So only our first and second statement is right. So option B is the right answer. Option B is the right answer. Okay, Q10 is not there. Pharmaceutical quality system. Q10 is pharmaceutical quality system. Next question is little bit lengthy but very easy from your parental preparation. As per Indian Pharmacopoeia, maximum number of particles per ml allowed for the large volume particles, parenterals, labeled as containing 100 ml or more of a single dose large volume injection intended for administration by intravenous infusion with particle size not exceeding 10 micrometer is. So basically they are asking if the particle size is not exceeding, if it is 10 micrometer, then how many particles or how many particles per ml are allowed? How many number of particles per ml are allowed? Option is given as 5 particles, option B is 25 particles, option C is 50 particles, option D is 10 particles. What is your answer? Very good. Option D, 10 particles per ml. Okay, because this is very important, you should see the size. If the size they have given 25 micrometer, then the only 2 particles are allowed for 1 ml, and that is the reason 10 micrometer 10 particles are allowed. Okay, so as of now option D is the right answer. Very good. The next question is the expert committee shall examine the report of serious event of death and give its recommendation to the licensing authority for the purpose of arriving at the cause of the adverse event within how many days of occurrence of the adverse event. So basically they are asking the expert committee should give their recommendation to licensing authority if so and so condition happened in case of adverse event. Within how many days they have to give this recommendation to the licensing authority after the occurrence of the adverse event. Okay, so they have to give their recommendation within 150 days, 30 days, 90 days, or 105 days. So basically, they will give the recommendation. The expert committee will give the recommendation to the licensing authority within 30 days if any kind of this adverse event occurs and related things. Once the adverse event occurs, from that day, in a period of 30 days, they have to give the recommendation. To whom they will give the recommendation? To the licensing authority. So option B is the right answer. So see here, event and the time limit is given. So ethics committee investigator reports any kind of adverse event serious, they have to report within 24 hours. If detailed analysis report should be submitted, whatever happened, the analysis report within 14 days they have to report. Expert committee, whatever their recommendation, they have to tell within 30 days. And licensing authority order compensation based on the expert committee recommendation. So these are the guidelines that they have to follow within this time limit. Next is according to new drug and clinical trial rules 2019, sponsor is required to forward his report on serious adverse event of death after due analysis to the licensing authority and the head of the institution where the trial has been conducted within how many days of the occurrence of the event. Now, here they are asking that according to this 2019 rule, Sponsor is responsible for conducting the study, any kind of whatever the clinical trial he is conducting. If any kind of death or serious side effect happens, he has to tell to licensing authority and head of the that particular clinical trial where the clinical trial is going on. So he has to tell within how many days. So option is within 1 day, 7 days, 14 days or 30 days. So the sponsor who is conducting the clinical trials, he should report to the licensing authority as well as head of that clinical trial where the investigation is going on. He has to report within 14 days. He has to report within 14 days if any kind of death or something happens. Okay, he has to report to them. The next question is, with regard to compensation in case of injury or death during the clinical trial, all of the following statements are true except. Now they are asking which of the following, basically which of the following is the incorrect statement. Which of the following is the incorrect statement? Financial compensation could be in the form of payment for medical management. Yes, this is a true statement. Our answer is not A. Option B is financial compensation is not provided for a child injured in utero because of the participation of parent in clinical trial. So if any kind of pregnant woman is being participated in the clinical trial and if something happens to the baby, then compensation is not given. Why? Because parent is involved. Is this statement true? No. The compensation is provided for the baby also if anything happens, even though only parent is involved. Okay, so ideally this statement is incorrect and this is the right answer. We'll see what is option C. In case of death, there is a provision to give financial compensation to the nominees of the trial subject. Yes, if anything happens by chance uh, uh, to the patient, then the nominees will be there, right? So the nominees will get the compensation. Option C is the right answer. Statement is right, not the answer. Option D is financial compensation shall be provided if injury or death occurs due to failure of investigational product to provide intended therapeutic effect where the standard care though available was not provided to the subject as per clinical trial. Yes, definitely their financial compensation will be provided in all the cases, but this statement is telling that it is not provided. This is the incorrect statement. Okay, option B is the right answer. So next question is, in scale up and post approval changes, that is SUPAC level 3 changes indicates what? This was also asked in the previous year. Option A is changes that are unlikely to have any detectable impact on formulation quality and performance. Option B is changes that could have significant impact on formulation quality and performance and usually require dissolution profile comparison in multimedia. Option C is changes that could have a significant impact on formulation quality and performance and required to conduct a bioequivalence study. Basically, the first option is there in the SUPAC, but it is not level 3, it is level 1. So it is unlikely to detect any detectable impact on the formulation quality, and generally there is no any requirement to prove any kind of dissolution, any kind of bioequivalence. The second option B is the level 2 where it is required little bit that at least a dissolution test should be performed to prove that they will have the same kind of dissolution profile. Option C is telling that there will be significant impact on the formulation and we have to prove that they both are bioequivalent with each other. So this comes under your level 3 where we will be conducting bioequivalence study. Changes that could have significant impact on formulation quality and performance lead to rejection. No. So this is level 1, level 2 and level 3 of the SUPAC. Okay. So level 3 option C is the right answer. Okay. The same things are given in the explanation part as well. So next question is, which one of the following procedure may be preferred by FDA to speed up the drug development process for drugs that are used to treat both serious and less serious disease, so that there are major advances in the treatment, provide a treatment where no adequate therapy exists. You know, in the COVID also it happened that there were no adequate therapies, and whatever the vaccines and everything has been approved, that has been approved based on the fast track only. Option is fast track, accelerated, priority review, breakthrough. So it has already mentioned that to speed up the drug development process. Okay, there are chances because either it is very serious, we don't have time also, like how the COVID situation was there. In such kind of situation, they go for the fast track approval from the FDA. Option A is the right answer. Next is the level of mercury as preservative in the cosmetics intended for use only in the area of eye should not exceed. So you know that phenyl mercuric acetate, right? Mercury containing compounds, they are widely used as preservative, but they should be used in very lesser concentration. So what is the concentration if we use in the eyes? 0.005%, 0.007%, 0.009 or 0.01%. So it should not exceed more than 0.007%. Option B is the right answer. Very straightforward questions are there, but remember, okay? So next is in India, IP reference substances are issued by, that is IPRS, Central Drug Research Institute. See, research is carried out here in the Lucknow, but they don't issue such kind of reference substances, right? So the reference substances are not issued by Central Drug Research Institute. Option A is not the answer. Chemical Laboratory Pune, there will be some analysis carried out, but they don't issue the reference substances. Option C is Indian Pharmacopoeia Commission, Ghaziabad. Option C is the right answer. Indian Institute of Chemical Biology, no. So option C, Indian Pharmacopoeia Commission, Ghaziabad, they issue this reference substances, and this reference substances will be further utilized for checking the impurities, quality of the product, assay and everything. So option C is the right answer. In other laboratories, the analytical method will be carried out, they will do everything, but not the issuing of the reference substances. Option C is the right answer. Next question is, which one of the following classes of clean air is required for aseptic operation? Which one of the following classes of clean air is required for aseptic operation? First of all, what is class 100? Class 100 is nothing but not more than 100 particles are present per cubic foot, and the particle size should not be exceeding 0.5 micrometer, 0.5 to 5 micrometer. So that indicates that is and class 100 is for the aseptic operations only, because very less particles are allowed in that one, rather it should be less than 100 only. Option A is the right answer, class 100. Next question is, which one of the following antioxidant compound is used in the aqueous system cosmetics? Option A is sodium bisulfite, option B is hydroquinone, option C is alpha tocopherol, option D is propyl gallate. Remember, alpha tocopherol, propyl gallate, that's hydroquinone, they are your oil soluble, and whereas sodium bisulfate, sodium metabisulfate, they are your water soluble antioxidant, and hence they are used in the aqueous system. Remember, these are oil soluble. Apart from these oil soluble antioxidant, you also have butylated hydroxyanisole and butylated hydroxytoluene. They are oil soluble. But when it comes as sodium salt, because it is a salt which is readily solubilized in water, it is used in the aqueous system. So option A is the right answer. The next question is, which one of the following application form is applicable in case of manufacturing for sale of Ayurveda, Siddha or Unani drugs. Option is Form 25E, 24D, 26D or 25D. Remember, for Ayurveda, Siddha and Unani system, there is application form 24D. Option B is the right answer. 25D is for the license which is granted. Okay, immediate numbers are there: 24, 25, 31, 32, 31, 32, 24, 25. 27, 28, same it is there. Immediate numbers are there for the license, and for application one number less. So for Ayurveda, Siddha and Unani, 24D is the right answer. Now the next question is, which one of the following guidelines are followed for analytical method validation if the method of analysis is not official in any pharmacopoeia or appear in any other recognized references? So whenever we are developing a method for a drug, whether it is HPLC or any kind of method, if any method is not available for that particular drug in the pharmacopoeia, then which guidelines we follow, right? So you know that there are ICH guidelines, and generally for bioanalytical method development, we follow ICH 10 guidelines, okay? So ICH, WHO, GLP, HACCP. So basically, ICH guidelines we follow for the method development if any of the method is not available in the pharmacopoeia. Okay, we follow ICH guidelines. Option A is the right answer. The next question is, which one of the following sections represent misbranded cosmetics under manufacture, sale and distribution of drugs and the cosmetics? Which one of the following sections represent that? Option A is section 9A, option B is section 90, option C is section 17C, option D is section 40. So very good, option C, section 17C is the right answer. So 9A is for the adulterated drugs, for the import of drugs and cosmetics. 9D is for the spurious cosmetics, for the import of drugs and cosmetics. 14 is for the confiscation for the import of drugs and cosmetic, and 17C is for the misbranded drugs or the cosmetic for the import of drugs and cosmetics. The next question is, which one of the following antimicrobial is used in the preparation of deodorant soap? Option A is bithionol, option B is propylene glycol, option C is trichlorocarbinolide, and option D is titanium dioxide. Very good, option C, trichlorocarbamylide is the right answer, that is used as antimicrobial, especially in the case of deodorant soaps. So you know that for to boost the preparation, we have certain important books for the drug inspector examination. So as you can see, drug inspector at your fingertips, where you can get all the objectives. Drug Inspector at your fingertips. Theory book is also available, plus Drug Inspector, where all the previous years papers are available. So make sure that you have these books where you can boost your preparation for UPSC examination, and definitely this will be going to very helpful for all of you, and you can order your books now and make sure that you have it with you, right? Next question is, which one of the following schedules in good manufacturing practices in India is used for the medical devices? Option A is M, option B is M1, option C is M2, option C is M3. So there are many, like M indicates manufacturing, but in that medical devices, M3 is there. Very important this also. Option D is the right answer. M1 is for the homeopathic, M2 is for the cosmetics, and M3 is for the medical devices. Option D is the right answer. The next question is, there is a requirement of self-inspection under GMP to be carried out by the licensed manufacturers of drug. A complete self-inspection should be carried out once in how many years? Okay. Basically, the complete self-inspection is carried out annually. That means once in a year. Option A is 2 years, option B is 3 years, option A is 1 year or 6 months. Remember, 2 years was for the initial where the retention period was there, but here the inspection, self-inspection should be carried out annually. That means once in a year. So option C, a year is the right answer. Next question is medical devices under the Medical Device Rule 2017 have been classified in which of the following classes? So the medical devices, whatever the devices like surgical instrument will be there, stents will be there. So they are categorized based on the like a risk. So it is categorized as A, B, C and D. A being the lowest and D being the highest. So option, it is not categorized as numbers like 1, 2, 3, 4. Rather than that, it is alphabetical A, B, C and D. So option C is the right answer. A, B, C and D. So here A is the low risk like surgical dressings, tongue depressor, etc. B is low to moderate like hypodermic needles, suction equipment. C is ventilators, bone fixation implants and etc. D is high risk like heart valves and everything are there. So when vital organs are involved, it is categorized as D. So remember the risk is like low to high, A, B, C and D. Next question is application for manufacture of cosmetics for sale or distribution is required to be made to the state licensing authority in which form? Form 3, 5, 6 or 7? Very good, 5 is the right answer. It should be given in form number 5. See, 3 is for permission to import or manufacture new cosmetics in India. Okay, either manufacturing or either if you are bringing cosmetics from outside importing, then also you have to go for form 3. 5 is for application to grant of a license to manufacture cosmetics for sale or distribution. 6 is for application for grant of loan license to manufacture of cosmetics for sale or distribution. And 7 is the self certificate of compliance of GMP for manufacturing of cosmetics. Likewise, you have 3, 5, 6 and 7. Next question is the premises license for manufacturing cosmetics shall be inspected by inspector appointed by the central government and state government to verify the conditions of license not less than once in 2 years, once in 5 years, once in 3 years or once in 4 years. So whatever the premises have been licensed for manufacturing of cosmetics, it should be inspected once in 3 years by the drug inspector which is appointed by central or state government. So you should inspect once in 3 years. So next question is, here now 1, 2, 3, 4 and Roman numbers 1, 2, 3, 4 are given. Penalty for offences committed without obtaining a valid license. That means there is no valid license for what? For manufacturing and sale of the drugs under the drug and cosmetic rule shall not be less than. So what should be the punishment? 1 year, 2 years, 5 years or 3 years. Okay, this is there. And fine should not be less than 50,000, 2 lakhs, 1 lakhs or 3 lakhs. So the years, that is 3 years and it should be not less than 1 lakh. Imprisonment up to 3 years and fine up to 1 lakh. So option 4 and Roman number 3. So option D is the right answer. Okay, I hope you have understood this question. Option D is the right answer. To maintain perpetuity of a license to manufacture drugs, application for its continuation is required to made within 3 years, 2 years, 5 years or 7 years. It is like an election. Election comes after every 5 years. Remember, it should be made within 5 years. Option C is the right answer. To maintain the perpetuity of a drug manufacturing license in India, application for continuation known as fee retention must be made before the completion of every succeeding period of 5 years. Before completion of 5 years from the date of issue of the license. Okay, that means if you get the license on 1st January, before that only, like before the 31st December, you have to make sure that you have to make the license and apply for the continuity of the license. So 5 years, remember the election comes in 5 years, every 5 years, like that you can remember. Chalo. Next question is, an Indian pharmaceutical company wants to market a fixed dose combination, that is FDC, where two drugs are mixed together and they are combined, of two drugs for an indication for which those drugs are already approved and given together, not as an FDC. Say for example paracetamol and aspirin. They are available as different. Paracetamol is a different tablet, aspirin is a different tablet for different condition. Now the company wants to give a fixed dose combination. They are already approved. They are already approved. It is there in the market, but as an individual tablet or capsule, not as a combination. The company wants to market it as a combination, but individually at the same dose. What will be the data requirement for marketing permission? So they want to say both are available as 100 mg, 100 mg. Now they want to give in combination at the same dose. The dose is also same. What will be the data requirement for manufacturing permission? Option is, marketing data to be submitted will be similar to data required for any new drug, including clinical trials. Will it be same? No, right? It won't be same because now you are mixing the drugs and then giving it. So option A is not the right answer. Option B is, chemical and pharmaceutical data as well as data on animal toxicology of new combination will be required. Let's see other options. Available data on pharmacological, toxicological, clinical data on the individual ingredients, along with animal toxicity and pharmacological data of new combination should be submitted. So this is the right answer. First you have to make sure that whatever data is available, because they are already marketed, their individual data is available. What individual data? Their pharmacological, toxicological, clinical data, that is available. Now extra study is that they have to give the animal toxicity and pharmacological data of the combination. Individually they are safe, but we don't know that after mixing they can give the reaction. And that is the reason option C is the right answer. Let's see option D, chemical and pharmaceutical data as well as data showing the stability of the proposed dosage form should be submitted. No. For us, important is individually they are safe. Are they safe after giving the combination? That you have to prove, and you have to submit the pharmacological and toxicological data of the combination. Okay, next question is, which one of the following schedules of drugs and cosmetic rules 1945 lists the requirement for the functioning and operation of blood bank for the preparation of blood components? So very easy trick is, you remember schedule F, F for food. Food after eating, where it goes into your systemic circulation, it gets absorbed. So F for food, and that goes into the blood bank. Very easy. M is for manufacturing, F is for the blood bank. Option B is the right answer. Remember all the schedules with the tricks and the mnemonics, it will be easy. Next is, all of the following statements categorize the drug to be spurious, except you have to identify which statement is not related to spurious. Option A is, it is manufactured under a name which belongs to another drug, that is spurious. Okay. Option B is, it bears or contains for the purpose of coloring only color other than which is prescribed. If it is labeled bear the name of a company which does not exist, this is also spurious. Option D is, it is substituted in part by another drug, that is also spurious. But what this option B is telling, that is not spurious, that is related to the adulterated drug. Okay, option B is the adulterated and not the spurious. So option B is the right answer. Next, consider the following statements regarding active drug transport. Now the question is from the biopharmaceutics. Transport of drug depends on concentration gradient. Active drug transport and concentration gradient, no. Concentration gradient is for the passive drug transport, right? So first statement itself is not this. Second is transport require energy. Yes, on its own it won't travel. Transport requires energy. Second statement is true. The process is capacity limited. Yes. And the rate is slower than the passive transport. Is the rate slower than the passive transport? No, right? So option only, there are 2, 3 options. C is the right answer. Next question is, the increased order of absorption from the available dosage forms are. So basically capsule is given suspension, emulsions and solution. So they are asking absorption. See, in the solution, the particles are already in the dissolved state. So there will be rapid absorption from the solution followed by your emulsion. Then suspension will be less and capsule will be very less, because capsule or tablet, they first has to undergo disintegration, then dissolution, then absorption, likewise, right? So they are asking increasing order. So capsule is less, suspension is little bit higher because no disintegration required. Emulsion is more higher and solution will be highest. So option A is the right answer. Very good. Chalo, next question is, which one of the following statement is correct for level B correlation in IV-IVC, in vitro in vivo correlation? It is a single point-to-point correlation between the dissolution parameters such as percent drug dissolved and percent drug absorbed. No, that is not level B, that is the A. It involves convolution and deconvolution technique. Level B correlation alone can justify formulation modification and manufacturing site changes. No, only level B cannot justify. It is a comparison between mean in vitro dissolution time, yes, and mean residence time, that is your MRT, in vivo dissolution time as per the statistical moment theory. So option D is the right answer, and it is not a point-to-point correlation. Point-to-point correlation is your level A. Okay. Chalo, next is, which one of the following properties is not exhibited by narrow therapeutic index or critical dose drug? What is narrow therapeutic index? So basically this is your plasma drug concentration versus time profile. This line is minimum effective concentration and this is maximum safe concentration. When this is narrow, we call it as narrow therapeutic index. Okay. Option is difference between the minimum toxic plasma concentration and minimum effective dose concentration is very low. Yes, it is very low. That is the reason we call it as narrow therapeutic index. We have to identify not exhibited. Okay. So basically, this is exhibited. This is the right statement. Option B is, it requires therapeutic monitoring based on pharmacokinetic and pharmacodynamic measures. That is also true because little bit increase in the concentration can lead to toxicity because it is very narrow. The range is very narrow. Possess very high within subject variability. Subtherapeutic, it does not possess high within subject variability. The variability will be less because if variability is higher, there will be difference in the toxicity levels. So this is not possessed by narrow therapeutic index. It is a broader, when the therapeutic index is higher or when the range is broader, there can be variation or intersubject variability will be there. Subtherapeutic concentration lead to serious therapeutic failure. Yes. Subtherapeutic means instead of this, your graph is below the effective concentration. So obviously, if it is below the effective concentration, you will not see any kind of effect. So there will be therapeutic failure. Option C is the right answer. The next question is, which one of the following is correct for drug administration via extravascular route at Cmax? Cmax is this, okay? And the time at which your Cmax occurs, that is your Tmax. Rate of drug absorption is equal to rate of drug elimination. No, then how it will be Cmax, right? It should be higher than rate of elimination. Rate of drug elimination is higher than the rate of drug absorption. No. Basically, here the rate of elimination is higher. That is the reason the graph is decreasing. Rate of drug absorption is higher than the rate of elimination. Yes. Because absorption is higher means more concentration is coming into the blood, and because of which you are getting Cmax. So it is higher. Rate of drug absorption is 0. If it is 0, you will not get any concentration, right? So at Cmax, rate of drug absorption is higher than the rate of elimination. Very good option. C is the right answer. Next is an applicant for grant of retail license in Form 20 or Form 21 or both is required to provide the premises which are in area not less than 8 meters square, 10 meters square, 12 meters square or 15 meters square. So the premises, the area should not be less than 10 meters square. Option B is the right answer according to rules 64 of DNC Act 1945, DNC Act and rules 1945. Minimum area which is required is 10 meters square. Okay. Next is the permitted synthetic organic, natural organic colors used in the cosmetic shall not contain more than. So arsenic is given, lead is given, heavy metal is given. What is the limit for them? So remember 2 ppm for arsenic, 20 ppm for lead and 100 ppm of heavy metals which are apart from the lead. Okay. So option C is the right answer. 2 ppm arsenic, 20 ppm lead and 100 ppm for the heavy metals. Option C is the right answer. Next is toothpaste containing fluoride should not contain fluoride more than. So what is the limit for the fluoride? Option A is 100 ppm, 500 ppm. Option B is 1000. Option B is 2000 ppm or option C is 3000 ppm. So toothpaste contains fluoride, right? So 1000 ppm option B is the right answer. Fluoride content in ppm and the date of expiry, everything should be there, and it should mention that it does not contain more than 1000 ppm. Next question is application for license to manufacture drugs other than those which is specified in schedule C and C1. Other than C and C1, it should be made to the licensing authority of the state in which form? 24B, 24, 24/7 or 24D. So as you know, application should be made in 24. Option B is the right answer. Other than C and C, C1 and X. If it is C and C1, it is 27. Next is license for retail sale of drugs other than Schedule X are issued by the state licensing authority in which form? 20, 21, 20B, 21B, 20F, 20G or 28, 21A. So it should be given the form 20B and 21B. Option B is the right answer. Remember, these forms are very important. You please go through it. So the next question is the drug specified under which of the following schedules to the drugs and cosmetic rules 1945 will not be sold by retail except on and in accordance with the prescription of registered medical practitioner. Schedule U, Y, H, H1 and X. So you know that list of prescription drugs, right? So that is see, Y is for the clinical trials. Anyways, that is not related to the prescription. H is there, schedule H, H1. Remember, X is also there because again that comes under your psychotropic substances. So X is also there. Ideally, it comes in 3, 4 and 5. Both are there. All three are there. Schedule H, H1 and X. Option B is the right answer. The next question is the medical devices shall conform to the standards laid down by WHO, US FDA, Bureau of Indian Standards, ISO, IC, pharmacopoeia standard where BIS standards are not available. So the medical device shall conform to the standards which are laid down by BIS, and if it is not available, then ISO and everything is there. So 3 and 4 is the right answer. Option D is the right answer. Very good. Next is, which one of the following is not applicable to non-linear pharmacokinetics? Non-linear pharmacokinetics means which is not proportional, which is not directly proportional. Okay. What is not proportional to what? Whatever your concept dose is there. Dose after absorption, it will give you AUC, the curve which I have shown you, right? So when it is related, I mean the rate of absorption is directly proportional to the dose, we call it as linear pharmacokinetics. But when it is not, we call it as non-linear pharmacokinetics. Now let's see the options. Option A is the elimination or absorption half-life change as dose are increased. It is not related to non-linear pharmacokinetics. The area under the curve increase in a proportional manner to the administered dose. This is not applicable, right? So definitely option B is the right answer, because the statement that option B is related to the linear pharmacokinetics and not regarding your non-linear pharmacokinetics. The composition or ratio of the metabolite of a drug may be affected by change in the dose. Elimination or absorption of a drug does not follow simple first order kinetics. Option A, option C and option D are related to non-linear, but option B is related to the linear pharmacokinetics. Option B is the right answer. Okay, now this is something that we have to solve. Okay, so here some data has been given to you and you have to solve it. Okay, one by one we will solve. First, write it down. What is given? Dose is there. What is dose? 500 mg. An IV dose of 500 mg is administered to a patient. It was observed that 70% of the dose was recovered unchanged in the urine. 70% of the dose is recovered unchanged. That means F is equal to 70%, which is nothing but 0.7. Next is what? AUC was found to be 362 mg/r per liter. Remember the units also, because here it is in liters and every option is in ml per minute. So we have to convert it. Based on this data, calculate TCR, MCR and renal clearance rate. So we have to solve one by one. So let's first solve the total clearance rate. You have to solve total clearance rate, right? What is the formula? You know that formula is nothing but dose divided by AUC. What is the dose? 500 mg divided by 362. Now calculate and let me know. So when you solve, you will get 1.38 liter per minute. You will get liters per minute. But as I told you, the options are not like per minute, it is in hours, right? Because AUC is in hours. AUC is given in the hour, so it is liter per hour. Options are minutes, ml per minutes, right? So you have to convert it. Total clearance rate is equal to 1.38 into 1000 divided by 60, because 1 liter is equal to 1000 ml, 1 hour is equal to 60 minutes, right? If you calculate, you will get 23 ml per minute. 23 ml per minute. I hope everyone is able to solve. Again, I'll repeat. Total clearance rate is equal to your dose divided by AUC. Dose is given as 500 mg and AUC is given as 362 mg into hour per liter. So this mg will go. If you solve, you will get 1.38. This liter, which is present as denominator, will go. So it will be liter per hour. But our options are in ml per minute. So you multiply 1000 divided by 60 because 1 liter is equal to 1000 ml and 1 hour is equal to 60 minutes. You solve it, you will get 23 ml per minute. Now in the option where is 23, both are there, A also and C also. Okay. We will solve the next one. As of now, this is not there, this is not there, right? Now next what is there? We want to calculate renal clearance rate. For that, what is the percentage? That is 70%. All which is nothing but 0.7, which is nothing but 0.7. What is our total clearance? So RCR is equal to total clearance rate into the fraction, right? So fraction total clearance is how much? 23 into 0.7. So you calculate this one. So approximately some 16 or 16.1 you will get 16.1 ml per minute. Now here you don't have to again convert because already we have taken total clearance into that is into ml per minute. Okay. So where it is there again in both A and C, in both it is there. Still we have not arrived to the answer. Chalo. Now what is the next that we have to calculate? Metabolic clearance rate. What is the formula for metabolic clearance rate? That is your metabolic clearance rate. 1 minus 0.7 into what was your clearance, earlier clearance that we have calculated? Total clearance is 23. So 1 minus 0.7 is 0.3 into 23. So somewhere around 23 into 3. 6.9 you will get. Right? 3 3 7 3 2 6. So 6.9. So option where it is there, option C is the right answer. I hope you have understood all the formulas for this one. Chalo. So C is the right answer. I hope everyone has calculated that. Next question is, which of the following statements are not correct regarding dissolution testing of solid dosage form? Option A is dissolution tests are used to aid formulation development, check product quality, performance, and to assess batch-to-batch quality of a drug with respect to drug product specification. Correct statement. We have to identify not correct. Second statement is dissolution test condition are not selected based on the properties of a drug and the type of drug form. It is absolutely not correct. We do select the dissolution based upon the properties of the drug. 2 is there. Dissolution is often the rate limiting step in the absorption of drugs with poor water solubility. Yes, that is the right statement. Dissolution cannot be correlated to oral bioavailability, while disintegration of dosage form is a strong indicator of oral bioavailability. This statement is not true. In fact, more than disintegration, dissolution is important because it can happen that drug is getting disintegrated, but it is not getting dissolved. Then you will not get absorption and there will not be bioavailability. So ideally, first and the third statement is true, and we have to identify incorrect statement. So that second and fourth is the incorrect statement. Option C is the right answer. Next is, which one of the following is not correct for dosage form prepared using protein based drug? Not correct. Trehalose and sucrose are ineffective in preventing protein denaturation. That is itself incorrect because they are effective as a protein denaturation. A is the right answer. Let's see what is option B, C, D. Low pH, that is pH 3 to 6, prevents chemical degradation. Yes. Cyclization of proteins makes them less susceptible to gut amino acid and carboxypeptidase. Yes. Metal gelatin agent can be used to enhance the stability. Yes, because they will not be free available to undergo degradation. Your option A is not the correct because they are very effective in preventing the protein denaturation. They are effective protein stabilizer basically. Okay. Chalo, next question is, which one of the following represents the correct sequential arrangement for a transdermal patch? So everything is given. Your from top to bottom, like this. So imagine that like this you are putting a patch. What should be the top layer? Like this they are asking. Okay. What do you think? First is the backing layer, that is true. First is the backing layer. Then comes what? Removable release liner, that is RRL. Next, what is given? Your drug reservoir, that means your drug will be present here, right? Next is your rate limiting membrane from where your drug will cross, and next will be your adhesive layer, right? Because you will remove this layer and then you will stick the entire patch onto the skin. Only in this option adhesive layer is in the D option, so D is the right answer. In the rest, RRL was given as the last, which is incorrect. Okay. Okay, so the next question is which one of the following represent the correct sequential arrangement transdermal patch from top to bottom in from top to bottom, right? So basically this is kind of a patch where different layers are present. You know that first is what? First is your backing layer, that is the topmost. Then you have the drug reservoir from where your drug has been loaded. From that reservoir, drug will be removed, right? Next is your rate limiting barrier or rate limiting membrane, which decides that how much amount of the drug is going to come at what rate and everything. Next is your adhesive layer, and next is your removable release liner. So basically this last step is nothing but we will remove this part RRL and then we will directly paste onto the skin. Okay. So this should be sequence BL, DR, RLM, AD and RRL. So option C is the right answer. The next question is inhalation aerosol should have particles which are porous with large physical diameter and small aerodynamic diameter. Oh, they should be smaller aerodynamic diameter. Okay. Porous with small physical but large aerodynamic. No. Non-porous with large physical and small aerodynamic or no. Basically, it has to be porous only with large physical diameter and small aerodynamic diameter. Next is in 2015, the formulation Spritam approved by US FDA is a 3D printed fast dissolving tablet, 3D printed transdermal patch, 3D printed delayed release oral dosage form, or 3D printed repeat action oral dosage form. Very good, that is the 3D printed fast dissolving tablet, and it contains levetiracetam as the active ingredient which is present, and it is based on zip dose technology. Okay. Next question is which one of the following statement regarding pharmaceutical powder is not correct? Hygroscopic powders absorb moisture from the air. Deliquescent powders absorb moisture from the air and partially or wholly liquefy. True. Efflorescent powder absorb moisture from the air and form water molecules of hydration or crystallization. This is incorrect because efflorescent molecules will release the water which is already present as a part of crystallization. So efflorescent means they will release, not absorb. Okay, option C is incorrect. And that is the right answer. Eutectic powders upon mixing become sticky or pasty or liquefy. Yes, because when they are mixed together, there is reduction in their melting point. So option C is the right answer. Next is which one of the following vehicles cannot be used for injection of drug parenterally? Mineral oil, corn oil, sesame oil, or castor oil. So you know that castor oil is like very thick and very viscous, which is not suitable for the parenteral administration, and we are not using castor oil. Option D is the right answer. Next is capsules are available in various sizes for human administration. Which of the following statement regarding capsule is correct? 000 is the largest capsule size. Yes, it is. 5 is the smallest capsule size. Yes. 0 is the smallest. No. 5 is the smallest. 5 is the largest. No. 000 is the smallest. No. So only option P and Q, that is option D is the right answer. Remember, 000 is the largest, followed by 000, 1, 2, 3, 4, 5. 5 being the smallest capsule size. Option D is the right answer. The next is, okay, now something is given here. 10 suppositories containing 300 mg acetaminophen have to be prepared using cocoa butter. Average weight of the blank suppository is 2 gram and the average weight of medicated suppository is 1.8 gram. What will be the actual weight of cocoa butter required to prepare the suppositories? Now you just write it down. What is the blank suppository weight? Blank is 2 gram, that means cocoa butter is 2 gram, right? Blank is 2 gram means cocoa butter is 2 gram. Then what else is given? They have given the medicated suppository. What is the weight of medicated suppository? 1.8 gram, right? And medicated means what? Your cocoa butter plus drug, which is 1.8 gram, right? In the medicated, your base and drug both are present, so it is 1.8 gram. And what else is given? 300 mg of acetaminophen is there in each suppository. 300 mg means what? How much grams? 0.3 gram, right? So in the medicated suppository, how much cocoa butter is there? Medicated suppository, cocoa butter plus drug is equal to 1.8 gram. Now we know that cocoa butter, we have to find it out how much will be required. What is the drug present? 0.3 gram. So this is the total weight of the suppository, medicated suppository, in which your 0.3 gram is the drug, 1.8 gram is the total weight of the suppository. From this we can find it out. Cocoa butter is equal to 1.8 minus 0.3, 1.5 gram. So 1.5 gram for each suppository cocoa butter weight is there. But they have asked for 10. So 1.5 into 10 is equal to 15 grams. 15 grams, option B is the right answer. You can also calculate the displacement value also from this one. Okay. So B is the right answer. Next is microemulsion have droplets in the size range of 100 angstrom to 5000, 100 angstrom to 1000, 1000 to 5000 or 5000 to 10,000. Option A, 100 to 5000 is the right answer. Next is, which one of the following is the best technique to sterilize thermolabile and moisture sensitive material? Both are there. If your drug is not stable to temperature and moisture, then which method you will use? Option is filtration sterilization, gas sterilization, dry heat or steam. Steam definitely not, dry heat we cannot, filtration because of the moisture we cannot, so we can go for the gas sterilization. We are using various gases like beta propiolactone is there, formaldehyde is there, so many are there. Option B is the right answer. Next is diffusion is facilitated in suspension via Brownian movement when the suspensions are deflocculated with particle size in sub-micrometer range, deflocculated with particle size in micrometer range, flocculated with particle size in sub-micrometer range, or flocculated. Diffusion is facilitated with Brownian movement when the particle size is less and when they are deflocculated, that means they are present as single entities. If they are flocculated, they will settle, right? Because there will be increase in the size when 3-4 particles come together, and that is the reason option A is the right answer.
Which of the following plant is the major source of reserpine? Okay, so you have to tell reserpine is a alkaloid, so you have to tell which is the major plant source of reserpine. Okay, Glemasium samprevis, Cantharus roseus, Colchicum autumnally, Rauwolfia serpentina. Okay, so I told you कि sometimes the trick behind to learn the pharmacognosy in better way is to link the link the plant material with the original plant source.
So 𝘎𝙎𝙾𝘼 actually from Serpentina, 𝘎𝙬𝙎𝑃 Serpentina. Okay. So 𝘎𝙎𝙾𝑖. Okay. So RES. See, this razor word is there. Okay. So from here you can guess. So 𝘎𝙬𝙎𝑃 Serpentina is the active source of 𝘎𝙎𝙾𝑖. Okay. 𝘎𝙬𝙎𝑃 Serpentina. So what are the synonym? Sarpagandha, Chandrika, Chota Chand, Indian snake root. These all are the synonym or what we can call common names. Okay, so common local names of these plants. So what are the biological sources? So biological sources of 𝘎𝙬𝙎 is consisting of 𝘎𝙬𝙎 serpentina. Okay, its biological source. What is its family? Apocyanaceae. The family of 𝘎𝙬𝙎 is Apocyanaceae. What is the chief chemical constituent? Is the 𝘎𝙬𝙾. Always they ask question about the chief constituent. The chief constituent of 𝘎𝙬𝙎 serpentina is 𝘎𝙬𝙾. Next question. Which of the following chemical is used in the preparation of dragon drops reagent? Tell me. Dragon drop reagent which is basically used. So dragon drop reagent or dragon drop test, it is the common test for alkaloid. Common test for alkaloid. Okay. So what are the reagent? Picric acid? No. Iodine in potassium iodide? Yes. Potassium bismuth iodide? No. Potassium mercuric iodide? No. So iodine in potassium iodide is the reagent, it's known as dragon drop reagent. Dragon drops reagent. Okay. Iodine in potassium iodide. So option B is correct answer. Okay. Wagner's reagent, potassium triiodide solution. It gives you reddish brown precipitate. Mayer's reagent, potassium mercuric iodide solution. It gives you creamy precipitate. Dragon drops reagent, potassium bismuth iodide. It provides you reddish brown precipitate. Hager's reagent, it provides you yellow precipitate. Okay. This you need to remember. These all are test for your two questions that has been asked from test from your one is from test of carbohydrate identification test, another one is from okay.
Starting with our pharmaceutical chemistry part. The question says spironolactone has which one of the following ring system? Is it gamma lactone, beta lactone, gamma lactam or beta lactam? So from the four options we can see spironolactone, that is a diuretic. We all know it acts on the collecting duct, and it says lactone, so that is clear that the structure contains a lactone ring. And if we see the C and D option, it says lactam. So gamma lactam and beta lactam. I guess beta lactam, you are familiar with that. The penicillins, okay, the cephalosporins, the beta lactam antibiotics, there we have a beta lactam ring, if you remember, like this. Okay, so we do not have a lactam ring here, but a lactone ring here. Okay, now what is lactone? Which type of lactone? Is it gamma or beta lactone? So basically we have different types of lactone. Now if we talk about our alpha lactone, it is 3-membered ring. Okay, 3-membered lactone ring. If we talk about the beta lactone, it is 4-membered. And if we talk about the gamma lactone, it is 5-membered. And if we talk about delta lactone, it is 6-membered. So here in spironolactone, we have a 5-membered lactone ring. So that means that we have a gamma lactone here. Okay, let me show you the structure also. So we have a 5-membered gamma lactone ring here, and that is called as our gamma lactone. Okay, so the B option, the A option is the correct one here. Okay, now coming to the next question, Gazette test apparatus is used for which one of the following limit tests? Heavy metals, iron, arsenic or sulfate? Like everyone, I guess, is familiar with the Gazette apparatus. If not, let me show you. So this is the Gazette apparatus we have. Okay, so basically we have what? We have a mercury chloride paper above. Okay, here attached a mercury chloride paper, and basically the staining, the yellow staining in that mercury chloride paper tells us The intensity of that yellow staining tells us that how much arsenic is present. So basically that staining is compared with the standard one, okay, the standard solution. So if it is more than the standard, that means the limit of arsenic is more than that is told in our pharmacopoeia. Okay, so based on that we have a gusset apparatus, and it is basically used for the arsenic. Okay, it is an used in arsenic limit test. For that we use a gusset apparatus. Okay, so I guess here the C option is the correct one, that is we use it for the arsenic. Okay, coming to the next question we have, benzene formation takes place during nucleophilic aromatic substitution, SN1 reaction, SN2 reaction, electrophilic aromatic substitution. So what is benzene? Benzene is basically a benzene ring with an triple bond. Okay, so this is a benzene ring and here a triple bond. So that is our benzene. Okay, now what is benzene formation and where is it happening? So basically what happens whenever there is our aromatic substitution happening. What type of aromatic substitution? Basically a nucleophilic aromatic substitution is happening. So that mechanism is also called as the elimination addition mechanism. Okay, that mechanism is also called as elimination addition mechanism. So what happens in that? Let's say we have an aryl halide. So let's say this is a benzene ring, we have an halide group attached here. Okay, now what happens in the presence of a strong base? In the presence of some strong base, that base can be anything, like we can say NaNH2, we can take liquid ammonia. Okay, all that are the strong bases. So in the presence of this strong base, what happens? This aryl halide, what it does, basically there is an attack of this strong base to the hydrogen that is adjacent to the carbon that is containing an leaving group. Okay, so it attacks an hydrogen there, and due to the attack onto that hydrogen, the elimination reaction occurs, and due to that what happens? That halide group is eliminated, and that results in the formation of our what? In the formation of our benzene. Okay, sorry, let me draw it again. Sorry, this is the halide we have. Okay, so basically here we have an halide group, and what happens? Basically due to the strong base, the elimination reaction occur, and due to that we get to see a benzene. Okay, now when this benzene is an intermediate here, further it undergoes what? It undergoes the addition reaction, and due to that addition reaction we have a addition of a nucleophile there. So that's why we are calling it as nucleophilic aromatic substitution. Means one halide group is substituted with some other group, and that benzene intermediate is formed there. Okay, so that is our basically nucleophilic aromatic substitution, and there our benzene is formed, that is an highly reactive intermediate. Okay, so the A1, the nucleophilic aromatic substitution, that is the answer here. Now coming to the next question we have, indole is synthesized by. Okay, indole, what is indole? I guess everyone knows indole. Indole is a heterocyclic ring, very well known heterocyclic ring that we can see. So this is an indole, and it is synthesized by what? So is it Sandmeyer reaction? It is Beckmann arrangement? It is Scarap's synthesis, or is it Fischer synthesis? So basically the indole is synthesized by our Fischer synthesis only. Okay, so what happens? We have a phenyl hydrogen along with ketone that is reacted to form our indole. Okay, that is the indole synthesis, basically Fischer indole synthesis, that also we call it. Okay, so Fischer is the reaction that is used for this synthesis. Next question, enalapril belongs to which one of the following esters of enalaprilic acid? Okay, so enalapril, have you heard about enalapril? That is a very widely used ACE inhibitors, the angiotensin converting enzyme inhibitors. Okay, that is our enalapril. So what is enalapril? Basically it is a prodrug, we can say. Okay, it is not in its active form. Okay, enalaprilate is an active form of enalapril. So why it is why we need a prodrug for that? So enalaprillate, it contains in its structure, if I show you, it contains 2 carboxyl group in its structure. It contains 2 carboxylic acid group in its structure, and due to the presence of those 2 carboxylic group in its structure, it is highly polar, and to reduce that polarity, it is changed to its prodrug, okay, by ester, basically ethyl ester. It is converted to an ethyl ester form, that is its prodrug, also called as enalapril, okay. So it makes it the balance of the polarity, okay, there. So that's why that is enalapril, okay. Enalapril is what? It is an ethyl ester of our enalaprillate. Okay, so if we come to the question, so enalapril belong to which one of the following ester of an enalaprilic acid? So is it methyl, ethyl benzyl, or N-propyl? So it is an ethyl ester, we can say, or ethyl ester form. Okay, here also you can see the structure. So we have the carboxylic group here. So one of the carboxyl group here is converted to the what? Ethyl ester. Okay, ethyl group is added on to that group or the carboxyl group. Okay, now coming to the next question, the reaction between a methyl bromide and the hydroxide ion to yield methanol is a SN2 reaction, SN1, E2 or E1. Okay, so let's try to draw it. So it is methyl bromide. So CH3Br, that is a methyl bromide, and hydroxide ion. Okay, they are forming a methanol. Methanol is what? A methane group. CH3OH, that is a methanol. So basically what is happening, a Br group is being substituted by the OH group, that is happening in the reaction. Okay, so basically it is not an elimination reaction, because in elimination reaction we will get a single bond from a double bond from a single bond. Basically an double bond or triple bond will be formed, but here we are saying the substitution is happening, so it is a substitution reaction. So it will be SN1 or SN2. Now if we see, now what is basically happening, that from this CH3, from one side the Br is being removed, and from other side the OH has been added. Okay, so basically that is the substitution is happening there, and this form is an intermediate form. So this reaction is following a concerted mechanism. Concerted mechanism means whole reaction is happening in a single step. Okay, so that happens in our SN2 reaction. Okay, SN1 reaction are the 2-step reaction, and SN1 reactions are the 2-step reaction, and SN2 reactions are the 1-step reactions. Okay, both the bond breaking and bond forming are happening in the same step. So that is SN2 reaction. Okay, so A is the correct answer here. You can see the transition state here, that is forming, okay, and methanol is formed here. Okay, now coming to our next question here, that is 𝘂𝐻 black tea is, 𝘂𝐻 black, everyone know that is an indicator. So is it a natural indicator, metallochromic indicator, redox indicator, or an acid base indicator? So 𝘂𝐻 black tea, that is a type of, we can say, whenever it is also called as, now basically 𝘂𝐻 black tea is a metallochromic indicator, means whenever there is a presence of a metal, okay, it reacts with some metal, it changes its color. Okay, so that is a metallochromic indicator, that is also called as complexometric indicator. Okay, so 𝘂𝐻 black tea is a metallochromic indicator, not a natural redox or acid base indicator, but a metallochromic indicator. Okay, next we come to the reactive intermediate in SN1 reaction is, SN1 reaction, substitution, nucleophilic, unimolecular reactions they are talking about. So these are the 2-step reaction, and in first step basically an intermediate is formed. Okay, so alkyl halide can be taken, alkyl halide, so that let's say CH3Cl only we took, okay, not CH3Cl, that is the least possible one. So let's say this we have taken, okay, so CH3, CH2, Cl. Let's say this I have taken, okay. Now reactive intermediate SN1 reaction. So let's say halide we have taken. Now substitution reaction is happening. So in the first step an intermediate will be formed. What is an intermediate? Basically this Cl group will be removed from here and a compound will be formed, an intermediate compound will be formed. So is it carbene, carbanion, free radical or carbocation? So basically in the SN1 reactions we have the formation of our carbocations that are further reacted with some nucleophile that results in the formation of one more different product. So let's say an OH is added here, so we can say OH. Like the example we took before, so that was an substitution nucleophilic, and there we make what? We have a formation of a carbocation. Okay, carbocation is an intermediate here. Like here also you can see we have a carbocation, and the first step is the slow step, that is a rate determining step, and second step is the fast step, okay, that do not have any involvement in the rate determination. Next, parameter that indicates polarity of a solvent. Is it dissociation constant, dielectric constant, boiling point or freezing point? Okay. So polarity of a solvent, the parameter that indicates the polarity is, we can say, the dielectric constant, yes or no? So the polarity is basically defined by the dielectric constant, like of water we see, or we can anything that the polarity we want to see, we see the dielectric constant basically, not a boiling point, freezing point or the dissociation constant. Okay, so dielectric constant is the basically the parameter that we check for the polarity measurement. Okay, now coming to the next question, the chemical structure of boric timolol has which of the following heterocycles? Timolol, a LOL drug, a beta blocker drug, we can say, a LOL drug we can say, yeah. So timolol. So we have a very nice trick to remember the structure of our timolol. So Ti and Mo. So from Ti we remember thiadiazole ring, and if we talk about the Mo, we remember morpholin ring. Yes or no? So morpholin ring. So in the timolol we have two heterocyclic ring. One is morpholin and one is thiadiazole. So the A1, the morpholin and the thiadiazole, that are the two heterocyclic rings we have in our timolol. Let me show you the structure. So this is the structure of timolol. It has an what? Aryl oxypropanol amine nucleus here. Okay, aryl oxypropanol amine nucleus here, and along with that it has what? It has two heterocyclic rings. Those are what? Those are the morpholin ring and the thiadiazole ring. Okay, that is timolol. Next question, the benzhydryl bromide is a starting material for the synthesis of benzhydryl bromide. What is benzhydryl bromide? So basically benzhydryl, if I tell you, so benzhydryl is basically like this. Let me draw it again. So like this kind of structure is called as our benzhydryl. Okay, now if they are saying benzhydryl bromide, so basically it is used for the synthesis of what? Diphenhydramine, diclofenac, diphenylhydrantoin or dipyrimidamole. So basically this benzhydryl group is present in our diphenhydramine structure, and benzhydryl bromide is an starting material for the synthesis of this diphenhydramine only. Okay, let me show you. This is the diphenmethyl, then we have our diphenyl bromide group here, then this bromine group, it is basically attached with this particular group there. So we have a benzhydryl bromide that is used in the formation or the synthesis of our diphenhydramine. That is a very widely used drug we can say, okay, diphenhydramine. Next, thyroxine can be synthesized from. Thyroxine, you must have heard about that, the thyroid hormones you must have heard, T3, T4. So thyroxine is one of them. So it is synthesized from what? It is L-tryptophan, it is L-tyrosine, L-pyroline, or the L-phenylalanine. So basically those in the thyroid gland, those thyroid hormones are synthesized like thyroxine, and they are synthesized with the help of an we can say the amino acid, that is L-tyrosine. Okay, so thyroxine, T3, T4, basically T4 and T3, they both are the thyroid gland hormones, and they are synthesized by the help of our L-tyrosine. Okay, so B1 is the correct one here. Coming to the next question, okay, Rochelle salt is, Rochelle salt, we use it many places, we use it even in the Fehling solution, if you know. Okay, Fehling solution also we have Rochelle salt. Fehling solution B, we have Rochelle salt with NaOH there. Okay, so Rochelle salt is also called as, is it potassium sodium tartrate, is it silver nitrate, sodium thiosulfite, or the potassium chloride? So basically Rochelle salt, the other name for that is sodium potassium tartrate only. So here we have is sodium potassium tartrate. Okay, that is the other name for our Rochelle salt. Next, 2,6-xylidine is a reactant for the synthesis of benzocaine, procaine, lidocaine, or cyanocaine. So basically 2,6-xylidine, so what is that? We have at 2 and 6th position, we have a CH3 like this. Okay, so that is our 2,6-xylidine group. And if we talk about that, what is it used for? So what it is synthesized, or it is used for the synthesis of our lidocaine. Okay, so it is used for the synthesis of our lidocaine, what? Our xylidine. Okay, this is the structure of our xylidine, and here we have a xylidine structure in the lidocaine also. Okay, so that is our lidocaine synthesis. C1 is the correct one here. Next, alpha, we have a max, okay, that is for the conjugated dienes can be calculated by which of the following rules or laws? So lambda max of the conjugated dienes, we use a formula for that, that was given by a scientist. So the rule or the law that we used for the calculation of that lambda max is it Woodward's law? It is Woodward's law. Is it Beer-Lambert law, Sedgwick rule, or Markovnikov rule? I guess that is a very obvious answer we have here. That is a Woodward-Fisher rule. Yes or no? So Woodward rule, everyone has studied that. So that is our lambda max for the conjugated dienes. We can use a Woodward-Fisher rule for the measurement or calculation of that. Okay, our lambda max of our dienes. So here we have a Woodward-Fisher rule. Next, which one of the following is used for the calibration of an absorbance scale of a UV visible spectrophotometer? Okay, the calibration of the absorbance scale. So basically for the calibration of the UV, the most widely or the most commonly used solution that we have is our potassium dichromate solution. I guess everyone is familiar with that. Whenever we study about the UV spectrophotometer, we study that for the calibration we use potassium dichromate solution only. Okay, so D1 is the correct one here. We use a potassium dichromate solution for its calibration. Okay, whenever we talk about the absorbance. Next, separation of ions in a mass spectrometer is according to their, okay, charge to mass ratio, mass to charge ratio, electron to proton ratio, or the proton to neutron ratio. I guess there is no doubt here. So basically in mass spectroscopy we have mass to charge ratio. Yes, everyone knows that. So mass to charge ratio, that is the thing that we have in our mass spectrometer. Okay, that is the principle on which it is based. All the spectrometer that is based on mass spectrophotometer. Next, the precision of an analytical method for an assay of drugs in the formulation within laboratory conditions like different days, different analysts, etc. is expressed as. So basically precision, it has different divisions, and they are specially asking for the condition of different days, different analysts, etc. can be expressed as. It is repeatability, it is reproducibility, robustness, or the intermediate precision. So if we talk about the different days, different analysts, so we have here is our intermediate precision. So let's see the different levels of our precision. We have 3 levels of our precision. First is repeatability. So we have same analyst, same instrument, same laboratory, same day. That is repeatability. Okay, that is repeating. Next, intermediate precision. We have same laboratory but different days, different analyst, different instruments. Okay, that is intermediate precision. That was asked in the question. Next is reproducibility. We have different laboratories. Basically, the laboratory is only different. Okay, in these two we are talking about the same laboratory, but in reproducibility we have different laboratory only. Okay, it is inter-laboratory studies. So that is the D1 here, that is like different days and different analysts that we have in an intermediate precision. Okay. Next, which one of the following detector is commonly used in FTIR spectrophotometer? Is it thermocouple, pyroelectric detector, 𝘂𝙎𝙎𝙎𝙎𝙬 pneumatic detector, or the photomultiplier tube? So 𝘂𝙎𝙎 pneumatic detector is also used in our FTIR spectrometer. Okay, but if we talk about, they are specially asking for the commonly used, so we have a pyroelectric detector, that is the most commonly used detector in our FTIR. Okay, if we talk about FTIR, most commonly is our pyroelectric detector only. Next, the non-aqueous solvent methylformide, dimethylformide, that is DMF, belongs to which one of the following classes of solvent? Is it aprotic? Is it amphiprotic? It is protogenic, or is it protophilic? So DMF it is non-aqueous solvent, as we know. So basically it is what? It is proton loving. Okay, so basically it is proton loving, so we can also call it as protophilic. Okay, so it is used for that. Basically it is used as an it is used to enhance the weak acidity of a weak acid. It is used for the enhancement of that, if we talk about protophilic solvents, and DMF is one of them. Along with that, pyridine is there, ethylene diamine is there. Okay, so that is also a protophilic solvent we have here. Next, which one of the following material is used in the construction of an attenuated total reflectance cell, ATRs? Okay, so ATR you must have heard about. So mostly diamonds are used in ATRs. Okay, because for the reflectance, okay, basically we use diamonds, they give the best results, but due to the cost of the diamonds, okay, it is very expensive, we use different material in that other than diamonds, that are much cheaper than diamonds. So if we talk about the options, germanium, sodium chloride, so potassium bromide or potassium chloride, so germanium is one of them. Okay, that is used other than diamonds. Okay, so diamonds are the most commonly used or the best used, we can say, but as the diamond, the cost is very high, so the alternatively we can use germanium also. Okay, that are the cheaper versions of that. Okay, so diamonds, zinc salides, germaniums, all that we can use in our ATRs. Okay, next, which one of the following is a reasonable internal reference standard for NMR spectroscopic studies of solvent in water of D2O? Okay, so they are specially asking for water of D2O. It is tetramethylsilane, is it silicon grease, is it silicon dioxide, or it is DSS? So basically, as in the NMR spectroscopic study, the standard solution, sometimes we also use that TMS. Okay, for the organic solvents we use TMS as a standard solution. Okay, but here they are specially asking for the water of D2. When the studies are in the solution of water of D2, they are specially asking for that. So in that case, the standard we take is at DSS, that is sodium dimethyl silapentane sulfonate. Okay, that is the thing that we used for our in the water of D2O, that we use as a standard. Okay, so don't get confused. You must have also studied about the TMS, but TMS is used for the organic solvents. Okay. Next, mercaptoacetic acid is a solution buffered when the ammonium citrate reacts to form a purple colored complex with. Okay, so mercaptoacetic acid. Mercaptoacetic acid, you must have heard about 𝘂𝙎𝙬𝙧 acid. Okay, so 𝘂𝙎𝙬𝙧 acid is also called as mercaptoacetic acid, and I guess everyone knows that in the limit test of our iron, there is a 𝘂𝙎𝙬𝙧 acid that is basically a solution, that is an intermediate that is formed. It is used there and it results in the basically formation of a purple colored complex with the ammonium citrate. Okay, that results in that. So which we can say limit test is that is our iron. Okay, so iron basically reacts with that. Okay, so it forms a purple colored complex. Okay, so that is our iron. 𝘂𝙎𝙬𝙧 acid, you must have heard about that. I guess everyone has heard about that. So that is the thing. We have a complex here of purple color. Next question we have here. What will be the concentration of our methyl testosterone solution in ethanol if its absorbance in 1 centimeter cell is 241 nanometer, was found to be 0.890 of methyl testosterone is 540 at 241 nanometer. So basically they have given us this is an numerical based basically, so we have to directly apply the formula of absorbance. Now here they have given different things. So they have given absorbance in 1 centimeter cell, so means they have given the length that is shown by B, so that is 1 centimeter here. And if we talk about the absorbance, they have given us to be 0.890. So absorbance is 0.890. Let's see specific absorbance. The specific absorbance they have given us to be in 1 centimeter is 540. Okay, so these are the things they have given, and we have to find the concentration. That we have to find. Okay, so what is the formula? Absorbance is equals to the specific absorbance in 1 centimeter, we can say, into concentration into length, or we can say B. Okay, so that is the formula we have. So what we have? We have absorbance. We have 0.890 absorbance in equals to, we have specific absorbance also, that is 540. Okay. Then we have concentration, no, we have to find out that, and then we have B, that is 1 centimeter. Okay, so 1 centimeter, we write it or not, that is the same thing, so we can directly remove it. So sorry, I rubbed it. Okay, so what was that? Absorbance is equals to the specific absorbance into concentration into length, where the absorbance was 0.890, absolute absorbance was 540 into C. Okay, B is 1, so we don't write it, that is also okay. So concentration can be equal to 0.890 by 540. That can be, that is how we can calculate the concentration. So if we do it, or if we solve it, so it will be about 0.0016, something like that comes about. Okay, so the most approx answer we get is as the C1. Okay, so you can calculate it using the calculator, or you can calculate it by yourself. So we get an answer like this. Okay, so that is basically directly in the formula we have to put the values and we get the answer. So the answer is our C1, that is the closest answer that we get. Okay, so C is the correct one there. That is the concentration we have, 0.001. In the assay of ferrous sulfate by titration against cerric sulfate using ferroin as an internal redox indicator, a red color of a solution is in titration vessel before end point is due to. Is it 110 phenanthroic ferric ion complex? Is it 110 phenanthroic ferrous ion complex? Is it oxidation product of ferrous sulfate or the reduction product of ferric sulfate? So basically, if we talk about the ferrous sulfate titration, the assay of ferrous sulfate by using the titration method of ferric sulfate, now they are saying they are using ferroin. Okay, now what is ferroin? Ferroin they are using as an internal redox indicator, and indicator plays a very important role. Okay, it is only responsible for the color change at the end point. Okay, so the ferroin is responsible for that. And what is ferroin? Ferroin is 1,10 phenanthroic ferrous ion complex, that is called as ferroin. Okay, so basically the color change will be based on that only. So B1 is the correct one here, that is it is based on the ferroin only, that is the internal redox indicator. Okay, so B1 is the correct one here, that is our ferroin, the 1,10 phenanthroic ferrous ion complex. Next question. The headspace gas chromatographic analysis is very effective technique for the analysis of a non-volatile compounds, mixture of volatile compounds, non-volatile compounds in the presence of a high proportion volatile compounds, volatile compounds in the presence of a high proportion non-volatile compounds. So basically, if we talk about the headspace gas chromatographic analysis, it is basically used for our volatile compounds in the presence of one high proportion non-volatile compounds. Okay, so D1 is the correct one here. Okay, it is used for the volatile compounds only, volatile substances only. Okay, next. In the assay of boric acid by aqueous acid-base titration, glycerine is added. Okay, so in the assay of boric acid, aqueous acid-base titration, we use glycerine. So why that glycerine is added? To dissolve boric acid, to enhance the strength of a boric acid, to suppress the ionization of boric acid, or to prevent the precipitation of a reaction product of titration. So boric acid is a weak acid, that we all know. Okay, so basically we form a complex of boric acid with a glycerine. So whenever that complex is formed, it basically strengthens the acidity, or we can say strengthens the boric acid. Okay, so basically glycerine is added to enhance the strength of our boric acid. Okay, so the B1 is the correct one, that is it is used as an borate glycerol complex that is basically making the boric acid, that is a weak acid, much stronger acid whenever it is in a complex form with our glycerine. So that is our
rough degradation of xylose. See, so we have during the class I have discussed you two things. One, I talked about Killian-Fischer synthesis. Another one is rough degradation. Okay, it name itself self-explanatory. Degradation means when a compound undergo degradation, rough degradation, so there will be one carbon less. That means xylose. Okay, xylose will be come into one carbon less. Glucose. So glucose is a 6 carbon compound, so it will not the answer. Fructose is a 6 carbon compound, not in the answer. Glucose is a six carbon, so here the triose is the correct answer. Because in case of rough degradation, when a carbohydrate or monosaccharide undergo rough degradation, one carbon will be reduced from the original structure. Okay, so triose is the correct answer. So next question. Formation of acetylcholine A is the link between which of the following process? See, chalo. So I will give you a little bit of description, then you have to answer. So so during glucose undergo oxidation, when glucose undergo oxidation, it will form pyruvate. Okay. So now this pyruvate is converted to acetyl coenzyme. Now this acetyl coenzyme may undergo complete oxidation to form carbon dioxide and water. Okay, this is the whole reaction. Now you have to tell acetyl coenzyme connecting which two pathways? Yes, answer this question. Glycolysis and pentose shunt pathway. Glycolysis या gluconeogenesis. Glycolysis या citric acid cycle या electron transport system. See, So conversion of glucose to pyruvate, basically your glycolysis. And is converse formation of carbon dioxide water, it is your TCA cycle or citric acid cycle. So it is the connection between glycolysis and citric acid. Very, very direct question they ask in the exams. Okay. You just need to read the questions and option properly. Chalo, next question. So this is the connection between glycolysis and TCA cycle. So all these things you will get in this comprehensive group, drug inspector in fingertips. So this is consisting of two books. One is theory, another one is your MCQ book. So if you want to read the theory, you can take this book. It contains all the essential information needed for the drug inspector exam. Second is your fingertip book, which contains all the important MCQ. So if you want, you will definitely, I will recommend it. You will have to definitely purchase this book and start reading from it. Next question. Vitamin B6 consisting of which of the following ring? Okay. So first thing is, what is the chemical name of vitamin B6? Comment down the answer in the comment section. What is the chemical name of vitamin B6? So vitamin B6 chemical name is pyridoxine. Pyridoxine. So pyridoxine contains which ring? Pyridine ring, piperidine ring, pyrimidine ring, yeah, pyridiazine ring. Tell me. Vitamin B6 consisting of which of the following ring? Pyridoxine basically consisting of pyridine ring. Please note it down. Very, very important question. Which ring it contains? It contains pyridine. Okay. Chalo, very good. Next question. So you can follow this chart. Vitamin A contains beta-alanine ring, cholecalciferol contains steroidal ring, tocopherol contains chromin ring, haloquinone, minoquinone, and vanadione contains naphthaquinone ring, thiamine contains pyridine plus thiazole ring, riboflavin contains isoalloxane ring, niacin contains pyridine 3 carboxylic acid, pantothenic acid, pantoic acid plus beta-alanine, pyridoxine, pyridine. This chart you need to remember, very, very important chart from exam. You can see how many time questions have asked from this chart. So that this chart has important information. You have to remember. Next, nitric oxide is biosynthesized form. Nitric oxide, NO. It is a vasodilator. It is a potent vasodilator. Okay. Which amino acid is the source for synthesis of vasodilator? L-citrulline, L-glycine, L-arginine, या L-tyrosine? Answer this question. Which amino acid is responsible for the biosynthesis of nitric oxide? Yes, I'm waiting for your answer. Which amino acid will respond? The correct answer is L-arginine. Very good. So arginine is the amino acid. It acts as a precursor for synthesis of NO. Okay. So option C is correct answer. Okay. L-arginine is the correct answer. Production of nitric oxide. The enzyme nitric oxide synthesis cleave the nitrogen from the 1-ethanol group of arginine to form NO. Okay. Chalo. Next question. Iodine value of oil and fats measures. Okay. Iodine value of oil and fat measures. Average molecular weight of the fatty acid, degree of unsaturation present in the fatty acid, free fatty acid, या unsaponifiable matter. What is iodine value of oil and fats represents? Tell me. What iodine value basically indicates? Iodine value basically measures the presence of free fatty acid in a compound. Okay. It measures the free fatty acid. See, iodine iodine number, it basically measures the content of unsaturated fatty acid. Saponification value, Richard Michel's number, and acid number. So you need to remember these are this check for purity of fats and oil. This test we perform for which purpose? We perform for to check the purity of fats and oil. Chalo, next question. So you have to match with the following. In type 1 globulin, you have high density lipoprotein, low density lipoprotein, very low density lipoprotein, and your glycoprotein. Next, you have carrier, your option 2, you have carrier of triglyceride from the liver and adipose tissue, collect cholesterol from the body tissue and bring back to liver, involved in the cell and lipoprotein interaction of lipoprotein, carries cholesterol from liver to the cell of the body. So you have to tell what is the function of which compound. So you know high density lipoprotein. Why it is called as high density lipoprotein? Because this has high density. And second point is it is also called as good cholesterol. You know why? Because it collect all the cholesterol from the cell and bring back to the liver where it get metabolized. So high density lipoprotein will collect the cholesterol and from the body tissue and bring back to liver. What is the function of liver? What is the function of low density lipoprotein? Because we called it as a bad cholesterol, yes or no? Because it transports all the cholesterol or transport the cholesterol molecule from the liver and it is and distributed throughout the cell. That is the reason the in chance of atherosclerosis is increased. Okay, so carries the cholesterol from liver cell to the body. So four. Very low density lipoprotein carries the triglyceride from the liver to adipose tissue and glycoprotein involved in the cell lipoprotein interaction and protein. So option A is correct answer. You just need to find out the correct answer. Okay, so you match the following type of question. This question was asked in UPSC DI 2022. Chalo. Next question. Which of the following method is not used for the separation of the plasma protein? Okay, not used. Flow cytometer, precipitation by salt out technique, ultracentrifugation, yeah, electrophoresis. Tell me, which is not used? Answer this question. Which of the following is not used for the separation of the plasma protein? You know, plasma proteins are present in where? In your blood. When you separate blood, you will centrifuge the blood, you will find plasma and serum. So these proteins are basically present in your plasma. So which techniques are used and which techniques are not used? Let me make you very clear. You know, centrifugation is needed. I told you that centrifugation is the principle through which these proteins get separated by the centrifugal process. So that's why this option is correct. Precipitation by salting out. Yes. So what we do, we basically precipitate this protein by salting out technique. So this option is also correct. Electrophoresis, yes, because you know we get separated the protein by electrophoresis because when we apply the charge, it will migrate low molecular weight will migrate faster and than the high molecular weight protein. So these three is you. But so flow cytometry is something different. So where you basically know the level of immune cells, immune cell infiltration. So that type of function you do by this flow cytometer. So option A will be the correct answer. Okay. So flow cytometer analyze and sort the cell, not the plasma protein, since it designed to chain analysis rather than the protein separations. Okay, so this is salting out technique, ultracentrifugation, and electrophoresis. These three technique is basically used for the separation of your plasma proteins. Okay, chalo, let's solve question from your microbiology syllabus. Okay, so first question: in indirect ELISA, a standardized monoclonal antibody is immobilized on the solid surface to support detection. A standardized polyclonal antibody is immobilized on the solid support for the detection. Antigen antibody complex is immobilized on the support for the detection. Standardized antigen is immobilized on the solid support for detection. So you know when we have ELISA discussed, so ELISA is multiple type. Your direct ELISA, indirect ELISA, sandwich ELISA, and multiple type of ELISA you have. So what we do in case of indirect ELISA? Indirect ELISA, what happens? So the on the coating surface of the ELISA tube, so antigen antibody complex is previously immobilized. Okay, for the detection. So here option C is correct answer. Okay, let's see. So indirect ELISA basically use detect antibodies in patient serum. A standardized antigen is first immobilized on the solid surface. If the specific antibody are present in the serum, they bind and immobilize the antigen. An enzyme labeled secondary antibody is then added, which binds to the primary antibody. After adding the substrate, colored reaction will develop. That intensity of the color is directly proportional to the amount of antibody present. Okay, so that is your indirect ELISA. Next question. Which of the following antibody is responsible for the allergic reaction? So very common question and very frequently asked question. When we talk about allergic reaction, we basically discuss about your type 1 type of hypersensitivity. So type 1 type of hypersensitivity reaction is associated with which immunoglobulin? Immunoglobulin E, immunoglobulin G, immunoglobulin M, या immunoglobulin A. Type 1 type of hypersensitivity reaction. Type 1 type of hypersensitivity reaction is actually associated with your immunoglobulin E, IgE. Okay, so option A is correct answer. Okay, IgE. It is produced by plasma cell located in the lymph node. It causes allergic reaction. It has shortest half-life serum, causes drug sensitivity, anaphylaxis type of reaction, immediate type of hypersensitivity, protect antigen from the worms, helminthes, and parasitic illness. Chalo, next question. Which of the following is not a bacterial vaccine? BCG vaccine, not. Okay. BCG is for which disease? Tuberculosis. Pertussis is for the vaccine for the disease whooping cough. Meningococcal, it is for the Neisseria meningitis. Measles for the measles disease. Okay, so measles vaccine. So BCG vaccine, it is a tuberculosis is a bacterial disease. Pertussis is a bacterial disease. Whooping cough caused by Bordetella pertussis. Meningococcal, it is caused by Neisseria meningitis. It is also bacterial. But measles is not a bacterial vaccine. It is a viral vaccine. It is a viral vaccine. Okay. So here option D is correct answer. Measles is a viral vaccine. Live attenuated viral vaccine. Okay. Chalo, very good. Next question. Which of the following statement regarding adjuvant is not correct? Yes. Which of the following statement regarding adjuvant is not correct? So first thing is, what is adjuvant? So adjuvants are the compound which is basically produced along with the vaccines to increase the activity of the vaccine. They increases the antigen specific immune response. They decreases antigen specific immune response. They preserve antigen integrity. They produce high affinity immune response. Regarding not correct, which is incorrect statement. They increases the antigen specific immune response. Yes, that is the reason. That is the reason why we are using, because when adjuvants are used, so they basically combined with form with the vaccine. So they basically increase the vaccine sensitivity. They preserve the antigen integrity. Yes, they produce high affinity immune response. Yes, but they decreases antigen specific. No, they do not decrease the antigen specific immune response. If they will decrease the antigen specific immune response, then how the vaccine will going to work? Okay, so that's why option B is incorrect statement. Okay, sure. So adjuvants basically one of the ingredient added in the vaccines for the stronger immune response. They act as a helper or booster for the immune response. They increases the antigen specific immune response. They preserve the antigen integrity. They produce high affinity immune response. Okay. Chalo, next question. Which of the following immunoglobulin are correct sequence based upon the molecular weight? Yes, based upon the molecular weight. You know, highest molecular weight. Highest molecular weight is IgM. IgM having highest molecular weight. Next come into the picture is next is your IgE. Okay. Next molecular weight is IgD. Next IgA and IgG is lowest. Okay. So this sequence is correct. Option D is correct. So you have to check IgG molecular weight around 150 kilodalton. IgM highest 900 kilodalton. Okay. IgA 160 kilodalton, IgD 180 kilodalton, and IgE 190. This chart is important from exam point of view. Everyone need to remember. Highest IgM, then IgE, then IgD, then IgA, then IgG. Okay. This sequence you need to remember. So this question was asking UPSC DI 2023. Chalo, next question. Consider the following statement regarding the vaccines. Polio Salk vaccine is live attenuated vaccine. Polio Sabine vaccine is a inactivated vaccine. Hepatitis A is a inactivated vaccine. Hepatitis B subunit is a contains HBC antigen. Following statement are you have to tell which one is correct and which one is okay. So how you remember? So polio, I told you polio have two vaccine. One is your Salk, another one is your Sabine. Salk is killed vaccine. Sabine is live attenuated. Salk is live attenuated? No. Sabine is inactivated vaccine? No. Okay. Hepatitis A is a inactivated vaccine? Yes. Hepatitis B contains subunit vaccine HBs antigen? Yes. So option D is correct. Okay. So these two statement is correct. Okay. Very easy. If someone remember about polio vaccine, they can easily answer this question. So this only needs your attention. Okay. Attention to the details to the questions. Okay. This is the complete chart. You can refer this chart to remember exact classification of X. Chalo, next question. Which of the following does not provide passive immunity? Inactivated microorganisms, humanized monoclonal antibodies, natural mother antibody transferred from the fetus, yeah, antitoxin, which will not provide humanized monoclonal antibody. Obviously, monoclonal antibodies are the see. You remember preformed antibody when preformed antibodies are produced. When preformed antibodies are produced, that actually provides you passive immunity. That provides you passive immunity. Okay. So how you will going to remember this? Humanized monoclonal antibody. Yes, these are the preformed antibody. This is used for the preparation. Mother natural mother antibody transfer to fetus, your IgG that directly transferred from mother to the fetus during through the placenta. This is also preformed antibody. Antitoxins are also preformed antibody, but inactivated microorganism, it is nothing but these are your killed vaccines. So when the vaccine what will come, so vaccine is not going to provide passive immunity. It will provide you artificial active immunity. Okay, what it is going to provide? It will going to provide artificial activity. So option A is correct answer. I want every students to write down each and every answer. You should comment down in the comment section so that it will it will be a proper practice of this model paper. Okay. Chalo, next question. Consider the following vaccine. The correct order of stability of vaccine is given. You have to give it. DNA vaccine, mRNA vaccine, live attenuated vaccine, and inactivated vaccine. So you have to tell which vaccine is you have to arrange it in the stability order. Okay. You have to arrange it in a stability order. DNA vaccine, mRNA vaccine, live attenuated vaccine, yeah, inactivated vaccine. So you have to analyze or you have to design in a stability order. So please remember these things. So DNA vaccine is most stable. DNA vaccines are the most stable. Next come is your inactivated vaccine. Next come is your live attenuated vaccine. And at the last, it, it will come mRNA vaccine. mRNA is basically less stable. Okay. The major problem, if you remember, the major problem associated with mRNA RNA vaccine is stability. RNA is highly unstable. They degraded if any sort of change in the storage. Okay. If its storage is minus 80, you have to store in minus 80. If its storage is -20, you have to store it -20. Slight change. If you just keep this vaccine for 5 to 10 minutes in out of the prescribed temperature, then the vaccine will start degrading. So that is the reason of the major major disadvantage of RNA vaccine is stability. Okay, so here the option 4, yes, option C is correct answer. Okay, you can see DNA vaccine is having highest stability. DNA vaccine is having highest stability. Inactivated vaccine, it basically contains your killed microorganisms. Okay, live attenuated vaccine, it contains live but weakened microorganisms. mRNA vaccine, it is highly unstable. It is rapidly degraded by enzyme RNase. So there is an enzyme whose name is RNase. Due to the presence of RNase enzyme, it basically degrades. And that's why mRNA vaccines are always stored in ultra low temperature. That is around -80 degree. We call it as a deep freezer. We call it, as them is less stable above the given vaccines. Understood or not? Students, tell me. So we have discussed the important questions from your uh, biochemistry, biotechnology, and microbiology syllabus questions that has been asked in the UPSC DI 2023 exam.
Now we will discuss the questions and their detailed explanations from the non-pharma portion. The first section in the non-pharma portion, the first subject that we are going to take is your computer knowledge, the computer language. There are many questions, around 5 to 6 questions that have been asked from the specifically computer language. So let's move on to this. So which of the following is not? The first question is they are asking which of the following is not an example of an utility software. So this was the question from DI 2023, and the main thing that they have mentioned that not. This not is very important, guys. If the question containing not, if the question containing false, if the question containing accept, incorrect, so these things become, now if you see these things, just become an extra conscious. Just read the question twice, because in these things, that's complete meaning of the question got changes. Okay. So what it is specifically asking, what is not an example of an utility software? So before moving things, the first thing that you should know what the utility software is. So utility software is what? It is a software that helps to maintain, manage, protect, and optimize the computer system. All these are known as utility software. So if you talk about what? If you talk about the option that is disk defragmenter. What is this disk defragmenter do? Disk defragmenter, it recognizes your fragmented data and improve the disk performance. So it is a type of utility software. Antivirus protects our computer from a virus, so it is also a type of utility software. WinZip, what is this WinZip do? A WinZip is an utility software that is used for file compression and extraction. But if we talk about this Google Chrome is a browser. It is not an utility software. It is what? It is your browser. So since they have asked which of the following is not an utility software, so the correct answer will be what? The correct answer will be your option number C, guys. Correct answer is your option number C. So utility software is a system software that helps maintain, manage, and protect optimized computers. So the utility software, all are antiviruses and utility software. WinZip as a utility software, disk defragmenter is utility software, but your Google Chrome is a web browser, that is an application software. It is not an utility. Okay. Next question, question number 138. Which one of the following programming languages is used to develop an mobile application, Android application for a mobile devices? So if we have to develop what? An Android application for mobile devices, so which of the following programming languages is used? First option is your Python, second option is your C, third option is your Java, and fourth is your C++. So if you talk about the Python, if you talk about specifically what? If you talk about specifically Python is what? Python is used for web development, data science, AI, and automation. If you talk about C, it is what? It is a system programming languages and operating systems. C++ is again, it is a game engine. Mainly it is used for development of games, game engines, and performance intensive components. But specifically if you talk about the app in mobile devices in Android, which language is used? They use Java. So the correct answer for this question will be your option number C. Okay. So Java is what? Java is traditionally been the primary programming language, and it is used for the Android application development. And the Android SDK software, that is software development kit, was originally designed to support the Java for building Android apps. If you talk about the Python, it is used for web development, data science, AI, and automation. Same thing with C. If you talk about C language, it is used for system programming and operating systems. C++ used for game engines and performance intensive. Okay. Which one of the following symbols represent the data flow in motion used for what? Used for data flow diagram, DFD. That is what? That is your data flow diagram. So which of the following symbols we use to represent the data flow in motion? If the data flow in motion, the first is your open-ended box, the second is your square, the third one is your circle, and fourth one is your arrow. So if we talk about the open-ended box, this open-ended box represents what? It represents the data store. It represents what? It represents the data store, meaning that data at rest. We have to identify that data flow in motion. So this will not the right answer. Square is what? Square represents an external entity. That is what? That is your source or a specific destination. So this will also not answer. Circle processes what? It processes the transforms of data. It represents if any data is transforming, so that particular process is represented by your circle. But if we have to represent the data flow in motion, that will be represented by your arrow. So the correct answer will be your option number D, that is arrow. So data flow diagram, DFD, is what? It is used to represent the flow of data within system, and arrow represents the movement flow of data from one process data source or external entity to another. Okay, next question. Question number 140. Which one of the following is designed to control the internal operation of the computer system? That is from UPSC DI 2023. If we have to control the internal operations of the computer system, the first option is your antivirus. The second option is your application software. The third option is your compiler, and the fourth option is what? The fourth option is your system software, guys. Okay. So if we talk about the antivirus, the main thing is don't only go for the right answer. If you know the right answer, just go with that. But if you don't know the right answer, or if you are a little bit confused, try to eliminate the option. If you solve the question from the elimination methods, the chances of questions getting true will always enhances. So if we talk about, since what they are asking, the question is asking to control the internal operation of the computer system. Simple. The first option is what? The first option is antivirus. What is the role of antivirus? Antivirus protects us what? From it is a utility software that is designed to protect us from the viruses. Protect our computer system from the viruses. So it is nothing more to control related with the control of internal operations. If we talk about application software is what? It is a specific app. It is a specific software. For example, MS Word is there, MS PowerPoint is there, MS Excel is there. So all these are what? Specifically application software. They have nothing to do with the what? Internal operation. If you talk about the compiler, what is the use? Compiler is used to convert the high level machine language. It is what? It is a language translator that is used to convert the high level machine language in high level programming language into what? Into a machine code. That is high level programming code into a machine code is converted by the compiler. So which option left is your system software? Yes, this is the right answer. System software is designed to control the internal operations the computer system. So correct answer for this question will be your option number D, that is system software. System software is designed to control the internal functioning of the computer system. It manages the hardware resources including the CPU, memory, storage devices, printers, monitors, and other peripherals. The operating system is the most important type of system software and act as an interface between the user, hardware, and okay. Antivirus, utility software, and all we have discussed. Okay. Next question. Which of the following performs the matching of domain names to IP addresses in the internet services? If we have to match the domain name to the IP addresses, which one of the following is perform this particular function? The first option is your HTTP, that is hypertext transfer protocol. The second one is your URL, that is what? Uniform resource locator. HTML, that is hypertext markup language, and DNS is what? Domain name system. So simple, what we have to, they have to perform the matching of domains. So matching of domains will be performed by whom? To IP addresses. That is what? That is DNS. What is the meaning of the DNS? DNS stands for simply your domain name system. So the matching of domain to the IP addresses is performed by using DNS. Correct answer is your option number C. DNS is what? It is an hierarchy and decentralized naming system that translates what? That translates human readable domain name into machine readable IP addresses. It enables websites like www.google.com and DNS act as phone book of the internet. HTTP is what? It is your transfer protocol for the communication between web browser and web server. World Wide Web is interlinked pages, and ICMP is what is used for network diagnostic and error reporting. Okay, next question. Question number 142. Which one of the following statements regarding the anti-spyware? Okay, anti-spyware is correct. Okay, so we have to find the correct statement. Anti-spyware. Okay, it edits and files that become not readable. No, nothing to do. It is used to analyze the network traffic. No, it is not like anti-spyware. So it is something related with the spyware. It scan all the files before they are installed in the computer system. No, it detects and removes. Yes, that's the measure. It detects and removes all the program installed illegally. What is this spyware means? Spyware means any program, anything that is installed without the consent of the owner. If it is your computer, it is your system, and anything that is installed without your consent, so that particular thing will be known as the spyware. So anti-spyware do what? It detects and removes all the programs that is installed illegally in the computer system. Okay, clear? So the correct answer for this question will be your option number D. That's it. So it detects and removes all the program installed illegally in the computer system, and anti-spyware is a software designed to detect and remove the spyware from the system. What is spyware? Spyware secretly collects the user information such as your password, browsing history, banking details without the user's knowledge. Okay. Next question. So before moving to the further question, let me introduce you guys with the Drug Inspector free batch test series. That is what? It is 100% free. Do you guys believe that? It is 100% free test series for all the drug inspector. Whether you have enrolled in our prime batch or not, it is absolutely free for all. What you will get in this? The first thing, that is daily practice set, that is practice daily, improve consistently, and you will succeed in exam. That is daily practice set test will be there, exam pattern based test there, performance analysis, detailed explanation. Okay, and it is useful for what? All state central drug inspector examinations. Okay, so how to get that? How to get that series? Just think, download the GDC classes app, and I hope if you are watching this video, you guys have definitely this GDC classes app in your phone, because over 500K downloads are already there. Now go to this batches section. In this batches section, what you will get? You will see this plus icon. In batches section, you will see this plus icon. Click on this plus icon and enter the batch code. What the batch code is? Batch code is GDCDI. This is the batch code. GDCDI is your batch code, and after entering the batch code plus on the OK batch, and you will be enrolled in this free test series for your upcoming drug inspector exam. Okay, so let's move on to the next question. What the next question is saying? Which of the following is not a threat to cyber security? Which one of the following is what? Not a threat to cyber security. They are asking the not a threat. Simple, not is there. So the first option is your denial of the service. Denial of the service, phishing, RSA, malware. So what is this denial of service? Denial of service do what? DOS, denial of service, overloads that particular website, so user cannot access this. So this is the threat to your cyber security. Phishing, what is this phishing? It tricks people and share this personal information. For example, a fake bank email that asks you what is your ID, password, and also this is also a type of malware. Malware is what? It is a harmful software. But if you talk about RSA, RSA is what? RSA is your reversed Samir Adelman. Reversed Samir Adelman. And what is this? This is an encryption method. This is an encryption method to keep your data safe. That is not a threat to software. Actually, it promotes the cyber security. So correct option is your RSA. Reversed Samir Adelman. It is an encryption method used to keep your data safe. It protects information by converting them into secure form that only the intended person can read. For example, sender, it types the plain text data, then writes your public, then it converted into the cipher data, and it gets into the private key. When it gets the private key, then it again decrypted into the plain data. Okay, that's all. Denial of service, it overloads a website with fake traffic, so real user cannot access it. For example, a shopping website crashed because of DOS attack. Phishing tricks people into sharing personal information. For example, a fake bank email asks you to enter your password. Malware, harmful software that damages a computer or steals the data. Okay, so this was all from the computer language, the computer knowledge questions. Now we will solve the questions from the English. Okay, so let's move on to the question number first. For the question number first, that is 144, is saying that which one of the following is what is a synonym of the word written in the capital letter. Okay, so the invaders who constantly raided India in the past and pillaged it were infamous for their rapacity. Okay, so the question is saying that they have capitalized a single word in a complete sentence, and we have to find what? We have to find their what? Find their meanings. Oh, sorry, find their synonyms. So the invaders who have raided India in the past, since we know that India is raided by multiple invaders in the past, so what, and pillaged it were infamous, so they were famous for their word. The word is rapacity. Now, if there is a question from synonyms and antonyms, so you have to, there are two ways to solve this. The first thing, if you know the meaning of this particular thing, if you know the meaning of this particular term, then you can correlate with this, all the options, and you can find it. But if you don't know the thing, then what you can do? Then you can eliminate the options. First, for example, the option is greed, criminality, exaggeration, and militancy. Criminality is what? Criminality means that the quality of being criminal, and militancy is what? Militancy is aggressive or combat behavior. So although these are the different things, but in this sense, if a person is a criminal or using the militants, so we can remove these options. Okay, we can remove these options. Now the next option is greedy or exaggeration. And if we go with the rapacity, what is the meaning of this rapacity word? Rapacity, the meaning of this word is your extreme greed. Why the invaders who invaded in India, what? They wanted our money, they wanted our gold, they wanted our things. So why? Because they were extremely greedy. So that the term, the meaning of this rapacity means what? Is extremely greed. And we have to give what? We have to find the synonym. So what will be the right answer? So the right answer for this question will be your option number A, that is greed. Greed is the right answer. Okay, so rapacity means what? Rapacity means extreme greed, especially for money, wealth, or possessions. It describes a strong desire to seize or acquire things unfairly or by force. Okay, so greed is the closest synonym of this rapacity. Okay, so these are the things that you can know. Criminality means what? The quality of being criminal. Exaggeration means what? Means overstating something. If you are think like the thing is small, if you have a small pain or anything, if you like things making like a larger, like if there is a small accident is there, but you tell something that there is a very big thing is there, so that thing is known as your exaggerating. Militancy is what? An aggressive or combative. Okay, next question, question number 145. What the question number 145 is saying that which one of the following is an antonym of the word written in the sentence. Okay, now what the sentence is: the impoverished children charitable schools were treated to an scrumptious meal by the local administration on Independence Day. Means they have seen that the children charitable school had treated a children to scrumptious meal by the local administration on Independence Day. Again, same thing that you have to do. Now options are, the first option is your unpalatable. Unpalatable means that the thing that is unable to eat, that we can't eat. Next is what? Next is your appetizing. Appetizing means what? Appetizing means delicious and appealing, means appetizing. Hearty, what is the meaning of this hearty? Hearty means again substantial and what? Substantial and satisfying. And scanty, that is insufficient. Scanty means what? Insufficient. So if we can see that this appetizing and hearty, appetizing means what? It is very delicious. Thing is, anything is that is very delicious or appealing, it means that it is an appetizing. Hearty means again substantial and satisfying. So in some manner these two are little correlated, so we can eliminate these things. Next one is your scanty and anapalatable. Again, what is the meaning of this scrumptious thing? What is the meaning of this scrumptious? Do you guys know? Scrumptious means that very tasty. Scrumptious means what? Scrumptious means very tasty or delicious. Very tasty and delicious. Again, tasty and delicious meaning what? And we have to find what? Antonym. Tastiest, delicious. Tastiest and delicious. Tasty and delicious means what? It is very good. But unpalatable, what is the meaning of this unpalatable? Unpalatable means that we can't swallow that particular thing. We can't swallow that particular thing. That means that very not tasty or unpleasant to eat. So this will the perfect antonym. What we have to find? We have to find the antonym. If they have asked the synonym, then the right answer would be what? Then the best possible answer would be your appetizing. So remember that if they have asked for the synonym, then it was appetizing. But since they have asked the antonym, so the right answer will be your unpalatable. That is option number A will be the right answer. Right? Okay. So scrumptious means very delicious, tasty, or appetizing. It is used to describe the food that tastes exceptionally good. Okay. But opposite meaning is unpalatable, which means that not tasty or unpleasant to eat. Appetizing means most delicious and appealing. Substantial means what? Substantial means hearty means substantial and satisfying. Next is what? Scanty means insufficient or meager in quantity. Okay, guys. Next question. Question number 146. This is what? This is one of the following. Which one of the following is most appropriate meaning of an item? Okay, so we have to find the meaning of this item. Means that break a leg. Okay, it is meaning that break a leg, to play a practical joke, to dance happily, to be violent with someone, to wish someone luck. Okay, so we know that these items and phrases don't have the direct meaning or direct meaning word by word. If we are going to do the meaning or find the meaning of this word by word, it means that we have to break the someone has broken any person leg. So no, it is not. To be violent with someone, no, it is not like that. So the meaning of the phrase is that is to break a leg is what? Is to wish someone luck. If that is to wish someone luck, to wish a good luck to someone. Okay. So break a leg is an idiom which is used to wish someone good luck, specifically before performance, speech, and examination. Okay. This is used in commonly like theater performing artists. We are saying that good luck was traditionally considered as bad luck. That's why they have started a phrase that is breaking a leg. Okay. Next question. Okay, so this was what? Consider the following jumbled word. Okay, these are the jumbled word, and which one of the following order creates a meaningful sentence? Okay, so we have to find the meaningful sentence. Okay, so if any question that have the meaningful sentence, if we have to arrange this, so we have to identify the parts of the sentence. Okay, the first part, that is A is himself, B is who, C knows, D succeeds, and E is he. So what is the subject here? So we know that the part of the subject, that is the subject, comes first. So what is the subject here? He. He is what? He is your subject. What is subject? He is your subject. So the first thing, and succeeds is what? Succeeds is your verb. He is what? He is your subject, and succeeds is what? Succeeds is your verb. And who himself, who knows, is what? These all are your relative clause in this sentence. These all is what? These all is your relative clause in this subject. So if in any sentence that contains subject, verb, and if any sentence contains the relative clause, so what happens? So here the subject will come. What will come? The first thing will be subject, then will be your relative clause, and at the end we have verb. Okay, so then we have relative clause and then have the verb. So what will be the right answer? First thing will be he who knows himself. Okay, he who knows himself. So E, then B. So first one will be E. Okay, so this will be not the answer. This will not answer. E, then who, B. Okay, so he who knows himself succeeds. Okay, so the correct answer is your option number D. That is he who knows himself succeeds. Okay. The sentence should begin with the subject. After that, there is a relative clause describing he, and main verb comes under that. Okay, so he who knows himself is succeeds. So the correct answer will be your option number D. Okay, next question. Question number 148. What is the question number 148 saying? Again, the jumble word. The first is perfection, manifestation of, is the education in man. Okay, so again, same thing. Subject. What is the subject? Here, subject is what? Here, subject is your education. Education is subject in this sentence. Like simple. What is the perfection manifestation? Is education. So education is the subject. After that, phrase. What is the phrase? Verb, phrase. Verb and phrase. So verb phrase is what? Verb phrase is your is the. It is your verb phrase. After that, verb phrase is there. Then noun. What is the noun in this sentence? The noun in this sentence is your manifestation of. Manifestation is a noun. That is the thing that person is doing. Manifestation is the noun, and that after manifestation, that second noun is also, that is followed by the perfection, and the phrase is what? The phrase is completed. That last, the phrase is your in man. The phrase that completes it is your in man. So what will be the right answer for this thing? The first thing that comes is what? The first thing will be your subject, that is your education. That is D. Okay, so all these are correct. Simple. If you identify the subject, if you are able to identify the subject, then you will be directly able to answer this question. So the what is the sentence? Education is the manifestation, education is the manifestation of perfection in man. What is the manifestation of perfection in man? Education. So education is subject. Okay, correct answer is your option number C. Okay, so education is the manifestation of perfection in the man. Simple, as we have seen this, guys. Okay, next question, question number 149. Which one of the following parts of the sentence that it is has an error? Okay, so we have to identify the error in the sentence. That is, it is widely acknowledged by a general census that whereas a failure is the stepping stone to success. Okay, failure is the stepping stone to the success. Okay, successive failures are not successive stepping stone into the success. Okay, the first option is your, it is widely acknowledged by the general census. Okay, simple. Oh, it is widely acknowledged by the general census. Do you understand what is the meaning of the census? Meaning of the census is the counting of people in that particular state or particular country. So do anything in the sentence have to do with the census? Do anything in the census that have to do with the census? No, it should be general consensus. It is what? It should be your general consensus, not your general census. What is the general consensus? Means general agreement or general common opinion. Okay, so it should be what? It should be your general consensus, not general census. Okay, so what will be the right answer? So we have to identify the error. So in which part of the sentence error is there? It is widely acknowledged general census, because it should not be census, it should be consensus. Consensus means general agreement, common opinion. So correct answer is your option number A. Okay, because census is what? Census means official counting of population. It does not mean public opinion. So correct expression is general consensus, which means general agreement or common opinion. Okay, so correct answer. Now, the last question of today's session. What is the last question saying? That again, we know we have to identify the error. We have to identify the error. The visiting student who had just arrived in Srinagar decided that they would navigate around the Dal Lake. Okay, so what is this meaning? That circumnavigate. So it is not meaning that what? Lake around the following day. Okay, so they have to identify what? They have to navigate around the Dal Lake. But the verb that is saying that circumnavigate, it is what? It means to travel all the way around something. Okay, but if we are adding the around, so we don't have to meaning this verb. No? So if this verb is added, the meaning of this particular verb is to what? To travel all the way around something. So around should is what? Around is an unnecessary repetition. So around should not be there. So what will be the right answer? The error is that the visiting student who had just arrived is right. In Srinagar decided circumnavigate around the Dal Lake. Okay, around what? Not should be there. So the word in which part of this has error? The around part. So the correct answer is your option number C. Okay. Okay. So the adding the word around means circumnavigate is redundant and unnecessary. Okay. Okay, guys. So with this, we have finished the complete paper of UPSC Drug Inspector 2023. I hope you guys have understood the concepts of not only the non-pharma, every subject. Okay, if you have any doubt, any question regarding any subject, you can mention that in the chat box in the comment section, and the faculties will answer that. And again, I will recommend you guys to not only focus on the questions and answers. Detailed explanations are there, so I will recommend you to focus on that also, because those are the explanation that will enable you, that will help you to solve the other type of questions also. Okay, so with this, we are ending the today's session, and if you have any doubt, again I am saying that mention that in the comment box, we will solve it. Okay, so thank you everyone, thank you for joining with